below 200 meters. Good morning, everybody. Good evening, good afternoon, good midnight, whenever you're listening. Thank you for joining us. This is the RV Falcor with Sebastian, and we are making dive 450. We are situated between Los Angeles and Catalina Island, diving to a depth of a target depth of 888 meters. Today's dive is going to be a little bit different than the others that we've had. Previously, we've been targeting hard grounds made of mineral-rich substrates, and today we'll be targeting sediments that have a different type of hard ground, a human impact hard ground, barrels of DDT production waste. There are a large number of barrels out there deposited in the 1970s, we believe. And our goal today is to characterize the microbes and animals associated with these as best we can, and ultimately to look at whether there are DDT concentrations or DDT derivatives that have accumulated in the tissues of the animals down there that are either on the barrel or in the sediments.
Can we turn off that?
We have
control.
a very specific sampling design set up where we will locate individual barrels and take paired cores in transects radiating out from the barrel in 4 zones. Adjacent to the barrel in a microbial halo that's been seen around some of these barrels, a meter beyond that, and 3 meters beyond that. The paired core design is an attempt to pair macrofaunal animal studies. We'll be looking at the composition and diversity of the animals in relation to DDT concentrations in the sediments with microbial and sediment characterizations. That's what the second core will be used for. So the Levin and Jensen lab will be combining efforts. We also will be taking some sediment scoops in an attempt to get larger animals and larger numbers of animals that we can use to assess DDT and derivative body burdens. You need quite a lot of tissue, and the small push cores we use don't generate enough tissue. So we'll also be doing some video trying to understand which animals are out there in this region, and over a kind of overview of what the purpose of this study is: to characterize the base of the food web, to understand how much DDT and derivatives are still out there in the sediments, whether they are making their way into the small invertebrates at the base of the food chain, and then ultimately others will be looking at whether those contaminants are making their way into fish and marine mammals and turtles and other animals in Southern California. It's something different for us, but this is pretty much an opportunistic sampling. There was a lot of media attention to these barrels, about 8 months ago or so with an LA Times article, and there's been some attention since, including a congressional hearing about this. So we're going to do our best to use our expertise to contribute to understanding what's happening right down there on the bottom.
Last time when they did the sampling here, did they find any other derivatives?
Yeah, DDT breaks down and it into DDE and DDD and other compounds, and so in fact much of it is now broken down, is our understanding. The concentrations aren't highest at the sediment surface, though. They're buried maybe 5 centimeters down.
And for sampling, what's your initial plan? I'm not holding it to it, but when we go to do push cores, is there a number of push cores you plan on doing at each site?
We're going to attempt do transects radiating out from at least 4 different barrels during this dive, maybe more, with paired cores in 4 zones. The first zone are directly adjacent to the barrel, then in a microbial halo that you can usually see a colored yellow or white, 2 cores, and then a meter out, 2 cores, and then 3 meters out, 2 cores. And then there'll be a few random cores, paired cores that would be where we see no barrels. It was suggested during breakfast discussion that so that we don't disturb the bottom, that you figure out where the current is going and orient facing into the current, so that if the current's going this way, that the sampling actually radiate out that way, so everything just flows away.
Sounds good.
Sampled perpendicular to the current, I guess. Yeah.
I can do that.
And we'll probably want to put each pair in together before pulling anything out, so that they're undisturbed.
So
8.
okay.
Total.
That's good. So
Eight-
each
8.
rack will be area if they're,
Well, that's good. We can have each rack be a barrel. Yeah,
and then
a barrel transect.
you got one odd one that only has 7.
We can use that one for the non-barrel cores initially. We have 4 racks of 8, right? Yeah. That's a good plan. Did we lose YouTube, I guess?
No, we've got it, but we can't see the chat messages on YouTube.
You want to do that same thing we did yesterday? Did you try that?
That's not going to work. That's not working. I don't understand it. YouTube claims that chat is disabled for the event, but nobody sees them chatting to each other and everything.
Go to the.
When I sit here for 5 hours, and then use my muscle.
Hit that arrow to the left of ROV dive. Yeah, hit that one.
So, it's just
Then go to the little 3 dots.
Yeah, I have to couple stretches.
Have you tried this one again? Get share of the link.
Oh, okay. No,
And then paste that into a new
I'm just
browser.
gonna have to roll out onto the floor. It'll be fine.
It's been like most of my adult life. Well, So do you think the YouTubers heard that discussion of what's happening?
I did. I do. I do. You can ask them. Double check.
Hey, YouTubers, did you hear the discussion of what we're planning to do down on the bottom today? Heard y'all. Thank you. Thank you, Lisa. Somebody said they didn't hear anything. Okay, a few people lost audio.
Should be okay. We've got 3 megabits per second.
No secrets. No secrets on the Falcor.
I think they're playing a joke now.
That is cute. What is it?
A glass squid.
Oh, beautiful. Are they related to glass octopus?
He has the same gold spots like that.
Huh.
Octopus did, but
Oh, that's lovely.
It's a lovely picture. I love the curly antlers on it.
You could make a coffee table book out of all these beautiful creatures. Bye.
He's like, as soon as it hit 90, he's like,
I'll talk about what we're planning to do again when we get to the bottom.
you gonna want me to have a look at these 3 or 4 barrels that are right here before I sit down? 'Cause I'd like to look.
And we're gonna want a Niskin.
Okay, they can
3.
hear us.
What's hole is on? Zoom out on yours.
Oh, yeah. I'm planning to be in another city.
That squid. One of the weird positions. Squid.
Sleeping squid total things.
Oh. What do you call that position? It looks like a yoga position. With the legs folded back.
Fold your legs back.
Oh, beautiful.
What's that?
You lean back and grab your ankles.
I'll do that.
Is that a cuttlefish?
I got his eye there.
J-Rod is so good at keeping our pelagic animals in focus and in view.
Oh, no, I like
And
this
this
one
camera is so amazing. So I wonder, was that a Humboldt squid, you think? It wasn't a market squid.
It was a pretty tiny squid. I don't know that it would be a humble, but had a different shape than
I mean,
used
it's
to
pretty
think.
deep. It's one of the squids that doesn't mind the oxygen minimum zone. Hey, Scripps Oceanography is tuned in to us. That's
Yeah,
cool.
we've got Woods Hole and Scripps. Both of them.
Hi, fellow oceanographers.
Or part of the barrel, or part of the barrel site.
Yeah, we hope you all caught our beautiful new seep site yesterday at Laswin Knoll.
Fish do not like this. Midwater fish, they make a lot of noise.
Yep. My understanding is that key elements of each dive are up on YouTube to review, so you can look at the older dives.
They eventually get put up to 4K.
It's 4K.
Eventually.
Oh gosh, there's one in Australia. I haven't got the data on board anymore, unfortunately, but we've seen so many.
So this dive will be a little different for us because it will be a sediment dive. We're descending with 39 push cores on board. This will be a record for me. I've been diving for a lot of decades. There they all are. People can see them, I guess, now. Yep. Don't think I've ever taken this many cores on one dive, and it's a little scary because it probably means we'll be processing samples all night. We have a couple of scoop bags as well, and our trusty suction sampler, but mainly we're focused on coring sediments and studying the animals, microbes, and contaminants in those sediments. Too bad we can't invite our viewers to come help us process tonight.
Sure, some of them would love to help you.
Yeah, I believe the slurp has been unclogged, correct? We got the rock out, we got the crab
Rocks.
out that was rocks.
Yeah, they're like hay rocks in there.
Oh my goodness. That's
Bring the
nice.
ISO down,
Which jelly is that, Devin? I know you know this one. It starts with a P, doesn't it?
It doesn't go down anymore?
Coralia. Does that sound right? Can you look it up? That's beautiful. I got one right. Rufescens. Yeah, I know the names of very few pelagic things, but
Yeah, we have to do an internal review
don't know if this is enough sheets, actually. We do one line per push core. But under there's four here. But under comments or observations.
Yeah, pretty consistent behavior for a while.
Well, I guess, where do we put the sample number? Under department?
Here. With the SCD number?
Yeah.
Yeah. So we'll have,
I mean, we can change all this.
and then you can see the little A, the
Oh.
SCD number.
The little number is the zone, we're all getting all the zones.
But is that going to be the SCD number?
So,
Okay.
look, wait, this is important. Where did I just put the? These are both A's. This is the zone codes.
So have you done any more research on this watch?
These are both C's.
Yeah.
These are both P's.
What you got?
We need to have a meeting?
I think we need to have a meeting.
Right, yeah.
Okay, we have a meeting.
Who are all the interested,
Every core is in.
who are all the interested parties that
But you're
are
also gonna have to write down the number.
so far?
So maybe we can have one of these columns be
We
the,
get 10
like,
a.m. meetings in on it.
like SCB
What do you
number,
think? You know, like,
and then push core
I
number.
wish I had a photo. You know that photo that's
Barrel
up in,
number.
purser office? Up at Balfour with that.
Is that okay? So that would be like SCB dash 256 is the first
I'm
one.
turning up
So
the-
the- And then the push core number would be 8. And the barrel number.
Yeah. Yeah. I mean, this is compartment, which is usually where we put the push core number.
Well, that's
but-
fine, if you want it there.
But as
You
long
can
as
change
you're
it.
okay that your form changes totally for this, then I'm gonna write down whatever you want.
You could put it where- whatever you think is easier. But the A's are the zone, they become part of the SCD number apparently. They ask for that. The zone, the A zone, the zone. And I need to make sure I write all that stuff down. So I guess we'll also time and depth. Depth is not So, one of the things we're also going to be doing that I haven't discussed yet is trying to ground truth Eric Terrell's survey. He was out here some months ago with some new instrumentation. I'm not really sure what it was, but he used it to locate what we think are barrel targets on the seafloor, and he's given us a map of many, and we're using that to help construct the dive, decide where we're going to go. But we're also interested to see whether his targets turn out to be barrels or of contaminant waste, or whether they might be other objects on the seafloor. So we'll be doing some ground truthing in that regard. This site is about 888 meters deep. So we're now less than 100 meters off the bottom.
Just start to get a return on the altimeter. We're about
So we will want to look around a little and off the bottom and take three Niskins. don't, I'm imagining it's pretty soft and smushy down there.
Okay. We like stuff, or taking.
Yeah, our bottom depth is gonna be 888, and the depth over here is 888. If you look at the dive plan. Look on the other side, it looks like that.
Should I show Chris this?
It makes it look like that's not the case, but look at the scale on the side. It's like not even
Chris, do you wanna see the barrel plan? Do you wanna see the barrel sampling plan?
Oh, sure.
Are you sure you don't wanna save your call when we land on the first barrel?
So they're paired cores, right next to the barrel. Oh.
okay.
Sometimes jellyfish get abundant just above the bottom. There's usually more organic matter in the water column and a boundary layer just above the sediment. So we see a lot of animals that hang out right pretty deep.
You're letting you get started.
No. Should be here. Come in. Oh, you want to Paul? That's good. Having Paul there.
Chris, can you point me out? Sorry.
You see targets down there?
No, I'm just, my winch stopped, so I'm yanking on my tether.
There it goes. Yeah, not sitting down might be difficult to get a good score in there, but like.
Is that a squid?
Singapore.
Oh, tuna pond. Oh, wow. Pretty.
Keep paying out a good speed.
Must be much smaller than it looks. A lot of quiet anticipation here in the control van. Everybody's excited to see what the seafloor looks like down here.
Playing here.
Well,
We don't usually have this many people in here in the morning at the beginning of the dive.
A lot of jellies today.
Yep.
depending on how the current is here
Is this the Borrelia as well?
It looks like it, but the tentacles look longer, so it might be a different species of Borrelia. I'm not sure.
Or a ton. They have some really, really long tentacles.
They are. They look like they're a meter long. It casts a wide net for food. I
Cast
guess they
net.
can feed.
Cast net of the deep. 50 off bottom.
We're very close to shore. Captain, do you know how far offshore we are?
Yeah, here we're about 6-7 miles off Santa Catalina, 7 miles.
7 miles off Catalina and a little bit further from the mainland near Los Angeles. But we can see shore on both sides of the ship. We can see land, I should say, so this is far inshore, very productive waters, lots of particles in the water column as we descend. That means there's lots of food here. And but what we haven't seen on this dive, which is very interesting, is few pyrosomes. Most days we've gone through a very distinct pyrosome zone in the upper few hundred meters of water. I didn't see them today.
They were at like 240 and not very many. In fact, maybe 10 of them.
Okay. So they were here, but not abundant. Yesterday we had a pyrosome party, a storm. Well, I think we called it a storm of pyrosomes everywhere, on the seafloor and in the water column.
You should be able to use the sonar. We'll use sonar and just stay consistent with it. Alright, I got one barrel on sonar,
Oh yeah,
possibly more.
possibly more. It's too bad we can't. Can we show our sonar on
You can.
on YouTube and Facebook? So we're less than 10 meters off the bottom now. Should be coming into sight. I see sediment. That's what we're expecting. I think I see a halo to our right. So there should be a barrel right near there, maybe. Maybe it's not a halo. Maybe it's a line of something. Fault line?
Probably not.
Is it something hard like a fishing line down there, or something that things are growing on, or a pipe? We definitely have sponges living on hard substrate. Some serpulid worms. Not a microbial halo. It's interesting though. I would like to know if all those white spots are snails. It's interesting. Not in the dive plan. But it's always good to take a look at what we have down here. There's a lot of that looks microbial to you, doesn't it?
Well, that's debris of some sort. I mean, if we don't see barrels, that's where it's happening.
There's a barrel not too far away.
No, we will.
Pretty sure there's a barrel not too far away. Start to see some bright blue stuff.
Clearly something that's been dumped over.
Might be one of those hoses, those discharge hoses.
Yeah. There's our pyrosomes.
Or maybe just a two-part dry look at this.
Oh, I'm trying to remember the name of the common snail that lives in the basins down here. I know it, but I forgot.
Returnal diseases, maybe?
Yep.
You could take that with your sonar.
This stuff is.
So that's more debris. Maybe pipes. or tubing.
All right, so good to go over where I think the barrel's at?
Yep.
And starboard.
Is Greg up? Hey Greg, do you remember the common snail that's all over the Santa Barbara Basin and our anoxic basins? I've collected a lot of them, but I'm not remembering the name.
There it is.
There's one.
You'll need a white balance as well.
Our first barrel.
Probably,
We're not seeing the microbial halo that we expected around it.
yeah, we'll just
Want some sponges?
get the quarters out.
You don't want the sponge. After the course, you want the sponge? We have section. We'd be at,
My preference would be a recent progress with the corner, but one of those sponges and.
we wanted to look at the DDT body burden and the vertebrates, so, yeah. I mean, how many other targets do you see?
Look, there might be one more out in the distance. Right there. When I get closer, I'll know.
Where did Paul go?
Right.
There he is.
Do a little count.
Okay.
So we're not exactly sure what was in these barrels, but you'll notice there's a big hole punctured in that one, and it's our understanding that to make these barrels sink, they did that intentionally, and so this looks like pretty good evidence of that. And so what we're expecting is that these barrels contain chemicals like DDT and their derivatives. It's not entirely clear that that's the only type of chemical that was dumped out here, but that's the one we'll initially be testing for. And so it didn't take very long to find a barrel. This was our understanding from Dave Valentine, who's been out here before. He told us once we're in this area that we're probably going to find them pretty easily with the ROV. And based on the mapping that Eric Terrell did and the number of targets that were detected, it does seem like there's an enormous number of these barrels out here, and it took us maybe less than 5 minutes to find the first one. And so we're just getting some images of it now, and we're getting some good coordinates, and then we're gonna take some push cores. And so this will be our first sampling effort of these barrels, so we're
So that we could count.
need to get our technique down, and we're going to try to do transects starting close to the barrel and moving away, and collecting a total of 8 push cores at each of 4 barrel sites to understand how the concentrations of any chemicals that might have been in that barrel vary with distance from the barrel. And so first we're trying to get some good imaging of the types of animals that are on here. It's possible that whatever chemicals were in here have been accumulating in these animals, and so we'll take some samples of them as well and see if the concentrations in the animals are higher than in the surrounding sediments.
Okay, so on the barrel, we're seeing sponges, serpulid polychaetes. It looks like some sabellid polychaetes, some Astris permodesta, that's a little snail. Paul, since there's no halo here, do you want us to just sample anyway?
Just do it. Yeah, barrel one.
Okay. Niskins first, however.
Yes. Did you do the Niskins?
No, but I've asked for them.
Thank you.
So we can see if they're pretty spread out.
Less meant for like just 5 years.
Oh, yeah, fair enough. Yeah. Might have to put it up
Jim,
somewhere
look at
bigger.
the Sonar, put it Sonar somewhere.
You want, you can put it in coat powder purposes. Alright, so Niskin first and then get the sample.
3 Niskins, please. These are going to be 2, 5, 6, 7, and 8. Is that correct?
Our first Niskin will be sampled 256, then 257, and 258. Yep.
I don't think we're gonna be able to get it. I'm still not even seeing this one down. I'm this far away.
Yep. Alright, there's pretty good there.
Okay, so I can get it. If you're down below. Just showing off to the right, off to the left.
Yeah, so there's something.
So you're trying to locate it in the sonar?
You have to get the
Just see
range
this-
just right for the sample so we can be consistent with our distance. So right now I have it, what's that first range range? 5 meters?
5 meters, yep.
Yeah, so that's us 5 meters away from it, 6 meters.
You happy with,
So, the
this
first
is
one-
the furthest away zone,
No,
but
what
we,
was the first one? What y'all wanted that?
next to the barrel.
Oh, we have to work our way into
We're
it,
gonna go.
so we don't disrupt it.
Okay. Have we got the Niskins already?
No, we
First
haven't
one?
done
No, what's the first range? So we're trying to figure out. 3 meters?
5?
5 total.
5 feet, roger that. And Niskin, do you want that over the barrel or just
Right where you are. Stay with me.
Here.
Roger.
If you want to sit down, it looks like Kira's going to give over her seat to you.
Paul? You still want us, if we're starting out and working our way in, you still want to be A, this to be A, the first, that's gonna be B. So our first sample will be 255D.
No, it's going to be 259.
We're starting out working in though. We're working out, working our way. So the way we have it now, the furthest zone is B.
We can do the closer one. Let me make sure fire first.
No, it's just a matter of us.
You believe that you'll create the least disturbance by doing that?
I think so.
I mean, do you want to put in all 8, or do you want to just do 2 at a time?
Not if we're having to move around. I think
What do you guys think? I would think that.
pretty far.
Work our way.
That's 5 meters away from
This
each
would be.
other.
Okay, so-
I mean, we might be able to do 2 sets.
256 and D,
We do
257.
one set right close to the ROV and then one set at like maximum.
Well, you can do them however is easiest.
We can do them what?
However is easiest for you. You want to pick up each move each time, do that.
Spotted building. This is some. Yeah.
That's all three fired.
Thank you.
Unless you guys have more. Then I got some current must be going to my starboard, so this is a good spot to not push for.
So we kind of have the current blowing.
To my starboard. So when I pick up any sediment, it should go off to my starboard instead of more.
Back on us.
No, it's gonna be a beautiful.
You don't have to be, but
They want us to build ships, which is okay with me.
all
It's
right.
okay with you.
All right, then it's really important that I just went through this with Kira, the SCB and letter number, the push core number, and our barrel number here, please.
Put it on high res.
Barrel 1.
Yes.
You know, there's a height there.
Alright, so you're just about this year, 256 was our first.
We need to rotate that sonar. Okay, so you said I'm 6 meters away?
Yep.
Alright, come forward a little bit.
258.
So we're 5 meters.
Well, currently we're at 6. If I come here, then
Put it down.
a meter out in front of us.
Do you think, let me just, before you
I
take a
just want-
sample,
Yeah, I'm
Paul,
gonna do
do you
another
really
test real
think
quick.
this
I wanna
is
see if
far
you can
enough
see it.
away for the far away
3
sample?
meters away?
This is 5
5
meters.
meters, that's what we talked about.
I know, but look at this. Like, for me, a
know it's
far away
5.
sample would be,
Then you put one, you can reach out
Distant,
here, you can
and
get
we're not.
5 and 4, right?
I,
Doesn't say, I don't know what, Dave's distances were.
Let's see how
I'll
it's
see what it is.
Doesn't-
I think what we wanna see is a gradient.
We're just having a discussion about how best to do this sampling, given that we need to be rather precise about our distances, making sure the current blows the sediments away. And discussing, how far out we think there could be a barrel influence and so on. This is a very old barrel. It's been punctured. We may not see any contaminant signal at all.
I mean, it stays like that.
We'll just give it a try.
So this is the 5 meters now.
It's gonna come out that far.
It's pretty far.
I'll try it.
You're on the bottom, aren't you?
Yeah. Oh yeah, I'm on bottom. Should stay
They
pretty
said their
solid
plan
right
is to work through the push core numbers in consecutive order.
They don't have to.
They're in groups of 8 as well. So the first 8 should be all
Yeah.
one barrel.
They don't have to be consecutive as long as we know what's what.
We'll try.
But
Yep. I'm
You're gonna
hoping
wanna
the.
You're gonna wanna set the 2 and let them marinate, or take
I
it
don't,
separate?
I don't think they have to sit for
no.
any length of
Push 'em
time.
in,
It's
pull 'em
just,
out.
make
putting
sure.
2 in so that
Yeah,
one
two is
just
not
doesn't
disturb
disturb
it.
the other when you
Okay.
pull it out. These should be well consolidated
I mean, in
sediments
theory,
that stay in the core.
you know, in theory, you could put one in and take it out, put the second one in and take it out. They both don't have to be in there next to each other.
But it's very common to have stuff fall out and disturb the adjacent spot,
Okay. Oh, yeah.
which is why we
Yeah.
like 2 together.
Good call. Excitement builds. Can feel it.
This is the most interest I've seen in push cores in a long time.
Okay.
This is a tried and true sampling method. I used this at the beginning of my career many decades ago, and the push core design hasn't changed a whole lot. It's basically a plastic tube, and probably the pilots can describe the way the sample is held in by a suction by itself.
I can do that. So just below the handle, where Chris is grabbing it at, you'll see a spring. It's holding down a washer with a rubber gasket. Underneath those rubber gasket, there's about 8 holes that go through that black piece that you're seeing at the top of the push core. As Chris pushes it into the sediment, the water pressure will actually push up against that washer and spring and let the water escape out the top of it, not compacting the sediment. And then when he's ready to pull it up, of course the spring pushes down on that gasket, creating a seal in the vacuum, and when he brings it up, it holds all the sediment in there.
Alright, you ready?
Thank you. Yep.
The camera out on that?
259D, correct?
You can go out just a little more. You can go out a little bit more.
Alright.
You want to leave things like that white stuff alone, right?
Yes,
Like,
if you,
you want like
preferably.
a somewhat clean,
that white stuff is a dead
and
pyrosome,
things like
so,
holes, ignore that as well.
yeah, I would say boring surface is what we're after,
surface,
no
gotcha.
specific animals.
Yeah, what are the white specks other than that?
White specks are fine. My guess is they're 4Ms.
Let's see, you are, looks like you're straight up and down.
The reason we might avoid burrows and decomposing animals is that they would change the microbial community and the animal community. So you can see there's already some disturbance, and that's the reason.
Yeah. So I don't know if there's a Guinness World Book of Records for a number of push cores on an ROV dive that you guys might want to think about.
If it's not this, I do not want to try to get that record.
Think about making this official, huh?
We still haven't got the Guinness Book record for the longest animal.
You do have it?
We should, but I don't think it's been verified yet. That siphonophore we saw in Australia, that was 150 meters long.
Oh my God, 150.
Yeah.
That was Christopher and myself.
Do you have a preference about the length away? Just
Like separation between the course,
Pretty close.
pretty close?
Yep.
Right.
about 10 centimeters from each other.
Closer.
Closer than 10.
5.
About that?
Sorry,
Yep.
the laser and all that.
It's okay. We basically would like to be able to compare these cores, assume that they're in roughly
Yeah.
the same contaminant regime and sediment property
Especially because you're taking this one
regime.
out first.
Yeah, definitely take this one first.
So 260D. This would be push core 2. And you're writing the barrel number down for us.
Alright.
Just do a little Cody wiggle.
So for those listening to us, we're labeling our core numbers by zone. D is the outermost zone away from the barrel.
Nothing.
You got it? Get compacted a little bit. 3/4 full.
Core looks good.
It's not dropping as much.
No, I just didn't have rotate.
You're right.
What's wrong?
Should we not have written it down?
I think this should be 2590.
be able to skirt a little bit closer to the bottom now.
You're right. Sometimes we turn off the mic when we're having an internal discussion about how to label our samples or how to collect them. So if you don't hear us for a minute or two, that's all. You're not missing anything important.
Or we're yelling at each other. We don't want you to hear that. Just kidding.
Not me.
Just kidding.
So now we're going to be 3 meters. We're going to be 2 meters out. Correct? Did our next sample is
So we debated how to do this morning. Our original thoughts were to start close to the barrel and work our way out, but then we realized that would disturb the sediment, and so now we decided it made more sense to start out about 5 meters and work our way in, collecting cores at 4 different points on these transects. So far so good. Looks like we're in a nice undisturbed area, and pilots are checking the distance from the barrel, and we'll have precise coordinates for each of these sampling sites, which we can use to also try to help us understand what these distances are. I don't know how precise the lat longe is actually, so maybe their measurements, your measurements are going to be our most precise for,
Sonar's gonna be,
yeah.
I mean, you
Yeah.
get a 1 meter accuracy.
Right, yeah.
Navigation. They say it's closer than that, but this is actually using.
Perfect.
to tell you how far.
Yeah, so we're using the sonar to judge the distance to the target, which is the barrel. And yeah, pilots are doing a good job with that, and so it looks like we're in a position to get the next set of cores.
We got those two. He's gonna put these up close to us, and he's gonna go out pretty much the distance of the arm.
Okay.
Put in the other two.
That's good.
The last two are gonna go right next to the barrel. And just while he's getting ready for this. So
No,
we don't.
Yeah.
how close to the barrel do you want? As close as we can possibly do.
For the
Yeah.
closest one.
Yeah, close with.
Rich.
Yeah, go ahead, Rich.
Yeah, just letting over a piece of on the green sea plant up here in the garage.
Okay, roger that. Can I have you move forward about like 20, 30 meters?
Yeah, look at that.
Yeah, so we don't know what's in these barrels. There's no way of knowing that. And that's one of the reasons we're sampling around this one, is it doesn't have a characteristic halo around it, which we've seen from some others. This may end up being a control barrel for us if we don't detect any toxic chemicals. But apparently there are many things besides barrels that were dumped out here as well, so we're also hoping to find some other objects, maybe of other shapes and sizes, that may give a clue as to what might have been inside of them. What we do know is that most of this dumping happened in the 70s, so these barrels have been out here for a long time, and you can see they're pretty corroded. We also know from prior work by Dave Valentine's lab out of UC Santa Barbara that the highest concentrations of the DDT and related compounds that they detected were about 5 centimeters down. And that's because the sedimentation rate's pretty high here. And so when these barrels were dumped in the 70s, there's now a couple of inches of sediment on top of them. And so we're expecting, once we get these cores on board, we'll section them in 2 centimeter sections going down the core, and we'll analyze each section individually. And we expect that if we do detect these compounds, they're going to be in highest concentrations, probably between the second and third section that we take from the sediment surface.
bridge.
Go ahead, Brent.
Hey mate, are you able to push the waypoints and stuff through again to us? We seem to have lost them all when we restarted the Green Seas laptop there.
Roger, does Falco waypoint come back in?
Yeah, Falco once we popped back in now, so we'll start creeping ahead.
Roger that, I will. Alright. You have
So,
more?
one of the things we would like to check is whether this location corresponds to one of Eric Terrell's targets. And
Yeah.
I don't know, is Jimbo, just curious about how we're gonna check whether this barrel corresponds to one of Eric Terrell's targets.
All the barrels are on this map, that targets.
Yeah.
So I'm off of
You're
where
not.
those were in that one, so I'm like in the middle of the patch, so we're a little bit off of what.
Okay. I think what
Not
I'd
far,
like to
though.
do after we sample this one is go to the target area where there's that cluster and just see if
Yeah,
we have something different, if we have halos.
I think absolutely we should try to
Okay, he
find.
wanted to sample here, so.
No, this is a good one. I mean, I've got no problems with this one, but yeah, we should definitely look for halos, look for other types of shapes.
I'm about 30 meters away from any of those targets.
So there was not a target here. Which is interesting.
Sometimes
No.
the things are off by 20, 30 meters.
That's what we
You
wanna
just have to
know.
find that offset. And so
Right.
That's a big option, though.
So this is push order for SCB 262 and C. Yes.
Okay, it fell out, guys.
Bring on the other side of the one that's in there.
Yeah, well, you think you can shake it out pretty clean?
Yeah.
And what we don't wanna do is disturb the next sampling areas.
Yeah. It's more to our right, man.
Well, you got the sensor sheet. I'm gonna give them a few with this one.
That's why
Yep.
we push them both in at the same time.
Yeah.
It's
Totally.
pretty soft. You can see it looks
See,
like
it's
jello.
very soft. Yeah.
Yeah, you can see the whole ground just shaking as I shake it out.
Yeah. Don't see any stratification.
Somebody's asking what we mean by halos.
Halo.
Dave Valentine's Jason crews photographed these barrels with what we think are microbial mats circling the barrels about a meter out or half a meter out from the barrel. They look like a white and yellow ring
You can almost see
around
it.
them.
You can see it just a camera.
In this one?
See the yellow? Over yellow patch?
Oh, look at that. Now we can see it. Look at that. You can see some microbial activity there.
Yeah,
And
so they
we're-
identified the bacteria. I forget what the major taxon was.
And so we're just calling those halos. It's not very distinct on this barrel, but there's a ring of bacteria.
Try to do instead of that.
I feel like maybe deeper down there's a little plug, harder sediment that could plug things up.
You just need like an inch of water at the top.
missed it.
You can see a oxidized zone about a centimeter down into the core. A little lighter brown means there's some oxygen penetrating into the sediments. Oh, no.
Get it in there.
If I'm in,
If there's enough, if we can get it in there.
we can probably use that if the upper part doesn't get disturbed. We don't have to have it so deep. I don't know how deep you need your core.
It's the top 10 centimeters we're really interested in, so I think this one looked okay.
I mean, if there's cores that you need to switch out, you know, we can take an even one and you can take an odd one if there's something. We can't, if it's vertically mixed, then we don't want it. But if the bottom falls out, that's okay for us.
Same situation for us. Vertical mixing is not good.
You know what I said, Leo?
So people who are watching, you're going to be seeing an awful lot of this same scene all day long. The less glamorous part of what we do, but very fundamental.
There's a lot of redundancy in science. So Lisa may have mentioned earlier, but not long ago, one of the Scripps ships with a team led by Eric Terrell came through this area and did some mapping. Because the bottom is so flat, it's possible to detect objects like this barrel that are sticking above the sediment using multi-beam sonar, I believe it's called.
He was using Remus, which is an
Okay.
AUV,
Right, they were.
and I assume that that's the instrumentation on Remus, some
Right.
kind of sonar, but
Some
I
kind
don't
of-
know. Is something new that he was trying that has a much higher resolution than the ship
Right.
multi-beam?
Yeah, so they towed this instrument behind the ship and collected data and were covered.
No, I think Remus is an AUV.
Oh, an AUV, autonomous, right?
Yeah.
Yes, so they don't have to tow it. These are some really sophisticated types of oceanographic equipment that people like Eric are developing at Scripps.
Remus might be a Woods Hole device, which might be why Woods Hole's watching.
Yeah, okay. Sorry.
I'm not
If anyone
sure.
wants to correct us on that, feel
I'm
free.
not sure. We really need Eric to weigh in here
Yeah.
and tell us what he was doing. But
And so Eric was
okay, so
Eric was able to detect thousands of these objects over a far greater area than anyone realized they had been dumped. And so with the data he collected, we were able to plot the targets that he detected and plan our dive and try to position us in an area where they look fairly concentrated, although that wasn't very difficult to do because there were so many targets detected over this large area. And so we expect that there are many more objects than he was able to detect. And you could imagine some of these targets may have sunk down enough in the sediment that they're not detected using sonar.
So those last two cores were also on some bacteria.
So on bacterial.
It's not a thick mat, but if you see this color out here, it apparently extends all the way to where we just poured.
Just note that it's on bacteria.
Some bacteria present, yeah, some visible on the sediment surface,
These were the push cores 3 and push core 4.
yeah. In other words, there's not a clear halo, there's almost like a zone right around here.
Alright, so do you want me to try to go for a specific spot just so the halos you want me to core into?
Yeah, I mean, it's fine a meter out. I think the bacteria are everywhere, but if you can see it, try to flip the core on the patch. If you can see a colored patch, the sediment surface. So what the ROV is doing is sampling zones, different distances from the barrel, and this zone, which we're calling Zone B, was designed to originally be on a bacterial halo. It's kind of diffuse out here, but we can see some coloration on the sediment that suggests that there's something microbial going on. Maybe it's just iron oxidation or something like that, but we're trying to target that.
It comes a little closer to us.
I mean, we're good. We wanted to be about a meter out from the barrels. That looks about right. Yep.
Well,
Hi, Ben. Oh, J-Rod disappears so fast. I don't even see him go.
whoa. That's something else.
Something hard down there?
No, something on that vehicle.
Oh.
Down with the sit loose. Yeah, that wasn't, see that was core number 28. That's fine, no, didn't do
What?
anything. We didn't do anything. I put my arm hit something on the vehicle right there. That's why we bounced it down. That's fine, it didn't affect anything. I bring camera back.
So occasionally you'll see 2 green lights. Those are the lasers, yeah. Tropic of Dan is on it. Probably knows how far apart they are. There we go, 10 centimeters. Yeah. The chat is very precise today. So very important to be able to measure the size of things because it's really hard to judge sitting looking at the screen. In fact, it's pretty surprising even with the laser sometimes when a sample comes up we'll be pretty surprised at how small it is compared to how big we thought it was going to be based on watching the video.
There's quite a few jellies floating around over the bottom. Pretty high densities, actually. If there is any kind of benthic pelagic coupling going on, those jellyfish are probably part of it.
Go look at some jellies. Science can't wipe things around.
Too bad we can't put a core catcher in the bottom. Would it help to move up? To move the ROV up to where, to the spot, make it closer?
It wouldn't hurt, but it's more it's just it's such a soft. I think I just gotta maybe a little deeper in there, maybe catch something more solid at the bottom. Let's see.
Very soft, keeps falling out,
Yeah.
which is tough.
Not much we can do about that except hope we find more places.
Yeah, I mean, we still want to target some bacteria, so you can maybe. Yeah, I'm just wondering if we can go closer so you don't have to travel so far back to the quiver.
No, unfortunately. Just to get
We'll get you water, I'll be back.
In there.
It's probably, like, for us, it's probably also okay to just tilt it slightly
Yeah.
to help.
Yeah,
help.
slightly.
I mean, I believe the integrity is still there, you know.
Got it more. Okay, I'll stop right there.
Thank you, Chris. It's good.
So it's SCB 264 C
264B.
B.
B.
Push core 6.
We're in zone B. And the next one's gonna be 263. Right?
Yes.
B.
B.
Yes. It's taking a while.
Well, this is good for us to see, you know, to gauge roughly the long it takes, and I mean, we're gonna try to do 4 barrels and some in betweens, right? Which I still believe we can do.
Yeah.
But it won't be up by noon.
No, that's for sure.
We can live with a shorter core if you can get it in. No, that's not gonna work. Okay.
Did you
It's really soupy. You can see it just go down and jelly-like. I don't know if it's because of the microbial.
It's all those pyrosomes.
I don't see too many. Well, there's
They're
one
good.
back there.
It's years of dead pyrosomes.
We'll take
It'll
it.
work if it
We'll take it.
if it stays. Oh, so long to travel.
It's gonna be hard for the back ones, you know.
see it?
It's in. All
Yay,
right.
thank you.
Well done.
Alright, so the next one, that paragraph is split up really close to the,
So they're-
wait, nice.
Yeah, like right next to the
So we have one more pair of cores from this, our first barrel site. And these will be as close to the barrel as we can get, so I believe we have to reposition the ROV, get a little closer. So coming to these sites wasn't originally part of our plan for this cruise. This cruise has been scheduled for a while now, and we've been working for over a week now on some nearby sites, but mostly looking at hard bottom areas, ones that are enriched in various types of minerals. And we've been characterizing the animals associated with those habitats as part of a NOAA ocean exploration program. And when word came out about how extensive this field was, we thought we would divert the ship and our original plans, and spend 2 days in this area sampling as well. It just seemed like an opportunity that we shouldn't pass up. The only type of sampling like this that's been done out here that I'm aware of was done by Dave Valentine's group, and that was using a different ROV, and it was about 10 years ago now that those samples were taken. And planning these kinds of trips, there's a lot of work involved, and usually ships are scheduled a year or more in advance. And so to be able to bring a ship with this type of ROV capability out here, the opportunities are are few and far between. And so since we were going to be here, it just seemed like it would be a mistake not to try and get some new samples from this area and increase our understanding of what may have happened here and what the long-term impacts of that might be in the 40 plus years since these barrels were dumped off our coast.
Hi, J-Rod, you're back. So we want to be like right next to the barrel, like as close as you can get without pouring the barrel.
Is it okay if it goes off to the right of the barrel, or?
No, it needs to be next. Oh, but I don't know what you mean by off to the right, but next to the barrel, right? Whoever's tempted to break open this barrel, it's already been broken open, probably.
Is that close enough right there, or closer?
What do you think, Paul?
Oh, that's perfect. Yeah, plenty
Okay.
close.
Thank you.
Very good.
Yeah. Is there such a thing as sponge cancer?
Not that anyone knows of. These little white spots are interesting.
For almost certainly.
Forams? Okay. It's coming out. That core is no good now. No.
Really soft stuff.
Yeah. It's tricky.
Want to answer that question on the last one?
About Remus, I can,
Actually, it was all right. Do they leave them in the so they can see them how close the two cores are on things? Hang on, I'll put the mouse next to it. The one I thought was the one. That one there.
why we leave them in when
Yeah.
we're sampling?
That exact reason?
We take two cores at a time pushed in because withdrawing one usually leads to sediment disturbance, and we're trying not to disturb the surface of the sediment in the vicinity of coring. You can see they're falling out a lot because the sediment's so soft and soupy, very high water content, and so to make sure that we have some clean patches of sediment to sample, we try to insert the cores together and then pull them out together. We don't know much about the effects of DDT on marine organisms of the type you're seeing here. We're not even sure this was a DDT barrel. There is no confirmation, so until we analyze concentrations in the sediment, we won't know.
One's looking better.
Here it goes. Maybe he can save some of it. No.
I think I said shoot, actually. Yeah, checking it out.
It's always a little bit painful to watch our sediment cores slowly slip out.
This is what I was talking about to the right of it. Is that good?
It's okay.
That's okay, I mean, I can go
Take
somewhere
what
else.
we can get.
We'll take it if it's gonna stay in the core. We'll take it.
No guarantee. Super soft.
No
Yeah, keep that
guarantees.
in mind.
Yep.
66A, core 8.
Or 8, yeah.
Adjust the heat. Need to adjust the heat. That's my one.
I wonder if you should go take the other one near it.
That's perfect core. Fantastic.
Great, thank you.
Yeah.
So I was trying to, I'm,
So,
okay,
Lisa-
Greg wants to wait. Yes,
So remember how y'all were talking about the sampling a little bit? What order we pulling out?
yeah,
Is it okay if we let them marinate, so we actually do put the first one in, put the second one in, and grab that first one first, maybe let it equalize a little bit better?
yeah, it doesn't, you can pick them up in any order you want, it doesn't really matter.
I think that might
We
actually.
just have a numbering system that will stick to you, it doesn't matter when you recover them.
Yeah, so if they grab the first one, it makes it easier.
What?
So if they put 2 in and grab the first one that they put in, that
Yeah,
would make it
yeah.
sequential for us and it'd make it easier too.
Yeah,
Make it easier for y'all too.
Thank you.
All right.
Find another one?
got the
Let's
first
fly.
date. Yeah, I think what we'd like to do is head over to the batch on our dive track. Okay,
Yeah.
That's where all the.
and do you have an event dropped right here, or could you just put a mark so that it'll show up there?
So we're gonna now head over to an area where a number of targets were detected with Eric Terrell's sonar survey.
Yeah, that's where I started.
This barrel didn't even come up in the data set that we had. And so we think there's a good chance that the extraordinary number of targets he detected is actually quite an underestimation of the total number of objects that are out here. And so we're gonna.
There is another explanation, that there is an offset with the Terrell coordinates, and that's what we're trying to learn, whether in fact they're 10, 20, or 30 meters offset in a particular direction. It was always a possibility.
Yeah.
Even though he found 27,000 targets.
That's crazy. I know.
That was just in your.
I know.
You left me at
It's
one.
amazing.
Oh, let's sneak into that corner again.
Yep.
Is that cool? Our weird video corner.
So we are, you said 30 meters off?
Yeah, so in addition to watching this live stream, I think there's also going to be videos and other media posted on the Schmidt Ocean Institute website. And all of this is being generated by Brady, our outstanding filmmaker, first time at sea doing this kind of work. He's more of a outdoor sports adventure kind of land-based filmmaker, and so
Not now.
he's now adapted to life at sea. And I'm not sure he liked it the first day very much, but He seems to have gotten his sea legs, and he showed us a video that I think's posted now on the site that gives a little overview of what our original cruise, why we came out here. It talks about our objectives for the cruise. As I mentioned before, this is a bit of a diversion for us. This is an opportunity to come visit this site that we didn't want to pass up. And so, yeah. So I don't know if you get proper credits on the Schmidt site for his work, but I was pretty amazed with a little video that he put together, sort of a synopsis of what we're doing out here, and it's very well edited and filmed, and so hopefully you get a chance to take a peek at that.
I'm just going to talk a little bit about the seafloor that we're traversing on our barrel hunt. There's a lot of what we see are sediment and burrow holes. Not really sure what is making the burrows and mounds surrounding the burrow openings. It would be a subsurface megafaunal organism. It could be thalassinid shrimp. It could be mollusks. It could be echeurin worms. We have a target. Looks like a piece of a cinder block or something blocky. Anyway, the bottom has a lot of pyrosomes on it. These are dead pelagic organisms that have fallen down, and they're in various states of decay. It's interesting that there aren't very many animals that have come to munch on them. Occasionally we see something eating them, but not often. And otherwise, they're really, we aren't seeing brittle stars or starfish or other fish. Looks like a few pieces of kelp maybe down here, but there aren't too many other animals, which is maybe surprising. However, I know our oxygen is super low at these depths. We have our oxygen plot up, and I'm guessing we're just a couple micromolar, and that alone could account for the paucity of animals. So we're in the patch and we're not seeing them. Is that correct? So we put the ROV in the middle of our targets, and we're now just looking around at whether there are any barrels here. There's a very nice kelp fall. Which, could I just zoom in on that real fast? Take a look at what's here. Now, if this was a better oxygenated piece of seafloor, we might expect it to be covered with pink urchins, maybe polychaetes, amphipods, but I'm not seeing much. Definitely has collected a few pyrosomes. Maybe there's some Asteris permodesta snails on here, but not a whole lot else. It's definitely decaying. We can see bacterial mat on top of the kelp. I can't.
Yeah. You want to go along the track line to find them, or just wherever you find them with sonar?
I think we'll try to use your sonar.
Yeah.
From here. There may be some kind of offset, or maybe not. There is a lot of kelp that we're done. Thanks. A lot of kelp that grows along Catalina Island and along the mainland. That's Macrocystis, and it makes its way down into the deep sea. We call that a kelp fall when we find it on the bottom. There's a lot of interest in kelp as a means of sequestering carbon dioxide in deep water. some of it goes into very deep water. If it's below 1,000 meters, the understanding is that that carbon stays out of circulation and helps remove CO2 from the atmosphere. So there are some commercial operations looking at massive kelp farms as a way through photosynthesis to remove CO2. Then they're looking at sinking that kelp into the deep sea. So there's a rock. Does that look like one of the...
There's a barrel. What is that?
Is that, I think it's a rock. I mean, not a rock, a cement block or some sort.
Cinder
Does it look like a barrel to you?
No. The cinder block.
See any other targets? This is sort of, we're using technology to try to identify what's effectively a needle in the haystack, except that there's a lot of needles out here. But still, it's not, 100% sure that each target we were given is actually a barrel. Here's another block. So I'm just wondering whether we should check out what these are. You want to?
For sure.
Let's take a look at it and see if it's a container of some sort or not. Since we know a lot of different containerized waste was deposited here. So we're past that cluster. Is that?
Okay, so no touching.
No touching.
I would no touching.
I can't tell what it is.
No, but it's something. Something that was dumped.
So that was the target that we had just ahead. Okay, so we're off. I guess we should
Yeah, we're gonna start
go
heading
back
this
to
way.
our track.
Yeah, Jason, good question. There's some articles in the LA Times that talk about this dumping, and one's more recent and one's from a few years ago. And I don't want to say anything incorrectly. There may actually be some lawsuits going on. I'm not sure about what happened out here, and so I don't want to speak incorrectly on that. It's kind of hard to imagine it would be legal to dump all these chemicals offshore, but who knows in the 70s
might have
if it's possible that there weren't any regulations against it. So you'd have to do a little research on that, and we'll be doing some of that as well ourselves, I suspect, to get a little better up to speed on the legality of what happened here. So I guess our bigger focus right now is just trying to find out what we're dealing with in the present time in terms of magnitude and effects and what sort of residual contaminants may still be here.
this a sponge? A
It's
giant
sponge.
bolt?
It's a sponge on something.
Looks like a giant bolt.
Like rock. Polymers for boat. That's a prop.
A boat prop.
No.
It's like
Propeller.
growling for it.
This is definitely the human influence dive. We can see, I feel like we always hear about urban seas. This is the urban deep sea next to a large population area with a lot of human input.
I'll say. So the pilots are using their sonar to detect targets and then guiding the ROV in that direction so we can get a sense of what they are. We also have a map that you can't see that plots targets that were detected by Eric Tyrrell's crews when he came through here using his sonar detection. So in some ways we're sort of ground truthing what he may have detected with what these objects might actually be. And we're also trying to sample around objects that we think may have contained chemicals. And so we clearly had one of those with that first barrel that we found. We've seen quite a few other types of objects that we don't know what they are. Clearly they're things that were dumped here. And yeah, now we're seeing something else. This white material is probably microbial, some sort of bacterial mat. That could almost be the top of a drum, hard to say. The sediments you can see if you've been watching the coring are very, very soft, so anything that was thrown over would have sunk in, and the sedimentation rate is quite high here. And so there could easily be 4 to 8 inches of sediment accumulating on top of things since they've been dumped, which makes them more challenging to find.
Something up ahead. I mean, could it be a corroded barrel where the sides have come off or something?
It's too distinctive in the middle right there.
I mean, I could pick it up and inspect it more if you want, but
It's another prop.
it could be just a used ordnance. It could be
A what?
used ordnance.
Ordnance. Military.
Thank you, training. You gonna touch it or no?
No.
Okay.
I'm just gonna put whatever to look back this way. Come back and check on it.
Well, thank you.
There's no reason to inspect those. They're not really what we're after.
I think getting the video is great, but yeah, I don't know if we want to do more than that.
I mean,
Yeah, so the total area is not known. The mapping covered a large area, and it didn't reach the end of the dump site, and so I think more mapping needs to be done. I know our Senator Dianne Feinstein was very interested in learning more about this, and a number of people from our institution and others gave some testimony to a committee, a congressional committee about this. Right now there isn't really any funding to study what's going on out here. Again, we're just here by chance. We had a cruise to work in another nearby area and just taking the opportunity to come here and explore a little bit. But I know Diane Feinstein is working very hard to try and bring attention to this and bring resources to allow people like us to come out here and get a better sense of the situation. And so, yeah, so I guess it remains to be seen how much actually gets done. Science doesn't just work without resources. We have labs to run and people to pay in those labs, and we need to be able to apply for grants to do our science. And so hopefully there'll be opportunities to do some of that. There may be some special calls through various agencies to come out here and study these areas. That's not clear yet. That may happen. That would be a good thing. Or we can just apply to more normal mechanisms of funding that we typically use to support our science, to see if we can generate some interest in studying these areas.
Are you going back to the other barrel?
Just to verify how
See what
big
it
it
looks like.
from a distance. So now we know what it looks like at a distance. We're gonna start going down that track.
Yeah, I mean, honestly, I'm surprised they're a little bit harder to find than I expected. So they're not that dense down here.
We're seeing a lot of objects. We've only seen one barrel. And my understanding is these ships would drop the barrels as they were moving, so they're probably going to be in a line of some sort. And if we can find one of those trails, then we might start seeing a lot of them. So it's just a matter of, finding that proverbial needle in a haystack on the bottom of the ocean.
I'd love to sample under the pyrosomes and find out whether there's animals attracted, whether a pyrosome fall changes the nature of the sediments and the kinds of animals that would live in the sediments or under it, or change the
Sure it
microbial
does.
community.
Yeah, sure it does.
A lot of little organic fall experiments down here, but that's not our mission today. Jimbo, what is the oxygen reading here?
Right now.
What is the reading? How low? It's very, very low, but I'm wondering what your... I mean, the absence of oxygen does affect how quickly material will degrade, and it also could affect the fate of DDT and its derivatives, the kinds of animals that might take it up or bioaccumulate. .46 micromoles. So that is super low. Just to give you some sense, there might be a few hundred micromoles of oxygen at the surface. So 0.46 tells us we're at way less than 1% oxygen saturation. And yet, surprisingly, the jellyfish are happy. But there aren't a whole lot of other animals down here. We're in a basin. I think this is the San Pedro Basin with a sill. Is that kelp?
Yeah.
Again, the fact that kelp is relatively undegraded. That's a feather boa kelp, egregia maybe. Okay, do you guys have targets at all?
Yeah, we have targets. There's just a lot of them end up being
Oh, there's another edifice that doesn't look like a barrel.
Oh, that's
These
looking
are
more
detected by the
ordnance-like.
sonar. There's one there and one there, maybe. Wonder if these are some of Eric's targets.
I would imagine
Oh,
so.
sure,
They're pretty hard
absolutely.
targets.
Yeah.
Yeah. So what we may have is a map of human debris on the seafloor, but not necessarily DDT contamination.
We also want to get a couple of random ones. We could do that at any time, I guess.
You want to stop and take them here?
Yeah, do that.
Or do you want to wait till we know that we're going to be sampling other, I don't know.
Really like to see some more barrels. These guys seem like they're in the middle of.
They're looking for
we
targets.
think
I
we
mean,
got one.
they're finding targets. It's just they're
got one.
I think you got
they're
one.
different things.
Look, there's a big. Looks barrel-like in the sonar. Let's see what they found.
Shadow.
Yeah.
If you look, so when the sonar sends out, pick up the metal and then throws a shadow behind it, so that's something big in front of us.
Yeah, so we got something big ahead of us.
I think it might be bigger than a barrel.
Yeah, there it is.
That's definitely
It's big.
A little round.
Oh,
found
uh,
seamount?
we found some geology. Kira, what are we looking at? I'm interested. Could we slow down and take a peek? What makes a big mound like this? Wouldn't be a methane mound. Is Kira here? Okay, so this was the target, correct?
Yeah.
That we were seeing. Got a lot of little sponges on it. It's a little bit strange, sitting right here in the middle of nowhere.
Yeah.
You know what these sponges are?
No.
Data waypoint first.
Yeah, Roger that. Will be now.
Yeah, Greg suggested we slurp these sponges as background fauna.
Okay.
And why not? We know there's been sponges on the barrels too, so we could compare. We were also talking about taking a couple of push cores that are not barrel related, so we might do that. Let's see if Paul's comes back. So these would be sponges on something on a hard surface, but it's not a barrel surface. So if we did want to compare to sponges on a barrel, we'd have a control.
So we're gonna take a slope.
Yeah,
We're gonna get images of it.
sure.
Camera is yours.
Not sure.
It's hard flying. So probably gets every time.
We don't know really exactly what these sponges are. Do I have focus?
We'll get focus now. We got focus.
Thank you. Not sure I'm going the right way.
We're in one. So all these blue guys could possibly be there. And we made them like this right here. And this condense.
We should do our background cores here too, if you're
Yeah,
doing a background.
yeah.
survey.
I don't think the little white specks are the asterisk. Greg asked for some of the snails, which are possibly on the rock. So we'd like, there's the snails. You see the little snails behind the sponge on the rock there? So we'd like to slurp some of them too.
That stuff right there.
These snails, right there.
There's the worm right there.
There'll be a variety of, there's a scale worm, very pretty.
So you ready to start?
Sure. So we'd like some sponge, snails, worm, slurp one.
Maybe the sponge last, or
What?
Say again, Joey?
I don't know, does it make sense to do the sponge last because they're
Yes.
kind of big?
They might clog. Okay, this is gonna be SCB 267, is that right?
Correct, 267. 267, slip 1. 267, slip 1, yes.
36.
Set up better.
Sorry, Paul.
It's all right.
I had to switch with you.
Did you?
I was struggling. I was struggling, man. I had to fly like this right here.
Thought I was going crazy.
I had to switch with you.
It's okay. Wait till you see. Wait till I do the hat switch later.
Okay. See what I can do for you, bud.
I could take the short chair.
I could,
If you want me
I
to.
could swap
It's
with you,
fine.
Paul.
No, this is makes me sit up straight.
There's that thing maybe.
Are we clogged?
No, I was just making sure there was one big rock that wanted to go in that river. It's not from us.
Piece of plastic.
Go through section.
Sectional.
Kira says this is just a mound of sediment. Maybe lithified. Let's see, hydroids. These little snails, Astris permadesta, live in anoxic basins typically, so they're very characteristic of low oxygen zones. We said the oxygen concentration here is less than 1 micromolar. Very, very low.
What would you like to call the strange possible? We could call it Ascent.
Get a few serpulids in there. I think we got the scale worm. I think so. Was back there, so it's not there anymore.
There's also.
Oop. We don't need the big blocky sediment bits.
These sponges will do?
Yep. If you, I don't know, do you think the big one will fit in there? Oh, easy peasy.
Like something off of Dr. Seuss.
Anything else?
Some of the, do you want some of this sponge right in front of us? Joanna? Yeah, some of this. I think it's the same thing.
I'll go in jar 1, right?
Right.
Scale worm down there too, so I'll get that.
Greg's favorite. Looks like one big piece that might actually block things. So I'm not going to recommend trying to take it. Is that okay, Joanna? We don't want to clog the slurp. What bio? No, in the bio box with the sediment scoops. We think we could. Do you want to leave it? Do you want to leave it or try to? I don't think it's going to cut off easily.
I would leave it.
We'll just leave it.
Okay.
We'd like to take 2 paired push cores in the vicinity. Our non-barrel cores.
Do you want it on the mound or?
No, off the mound, please.
You want to use those, the 7, the
Yeah,
rack of 7 for that?
from the rack of 7, sure.
How many are you going to want?
2?
Or
We're taking a couple of cores that are not associated with barrels as background comparison cores.
I didn't like it. Alright, so out here, is this fine?
That's great.
Yeah, Greg reminds me that Astris permodesta, that little snail we just sucked up, its new name is Alia permodesta. I did have a cruise on the Nautilus. I believe it was in San Pedro Basin where we saw these snails in the water column. I had never seen them lift off the bottom and sail, but they had stuck out their foot in a way that, yeah. Is this 33?
Yes.
What is EE?
It's 33.
Oh.
Paul, we have up to 39, so. And this would be 2, 6, 8, and 9, right?
268, which score EE, or 33, also known as 33.
And there's no letter after them, right?
No letter, correct.
Anyway, these snails can basically go parasailing in the water, and I think it's to get out of the low oxygen conditions. Not really sure what induced that.
Your arms can be all the way out, and some white bones and then square.
You're gonna work on
That white square.
where the splashing number.
Looks very soft here. See. Where did you see the radio interview?
Email.
I don't want to read my email. There's hundreds of them. My last email is downloaded at 6:53 a.m. So.
I'm in there.
Yeah, it's got a big one. That one worked a lot better.
It's a big worm. That'll be fun.
Oh, wow.
You can't tell if it's a polychaete or an emerdian. It might be a big nereid or something. But it's an even number and you get it. You're gonna give us the worm?
We can switch if you want.
Okay. I might make a note that there's a big worm in there. We're having a little discussion because the Jensen lab is getting even numbered cores and the Levin lab is getting odd number cores. This is just how we know which of the pairs we're taking. But when there's a charismatic animal like that big red worm, we have a discussion about trading.
Rock, paper, scissors. It's a
Something like that.
So worms are in the category of charismatic animals now.
Around here they are.
Okay.
They've always been charismatic to me.
Most people think whales, dolphins, turtles.
No, not in our lab. My roots are in worms. My thesis was on worms.
Yeah.
It's a little stickier. core is not so deep.
All right.
Thank you, J-Rock.
Better than last time. Okay, 268 is push core 33, 269 is push core 34.
All right, now what we need is to develop a barrel finding strategy here. I'm not sure these targets are particularly,
I mean, there's definitely a lot of hard targets.
I mean,
out there.
they are accurate in that they are hard, but they're not necessarily barrels, so I'm trying to think about how to find some.
To go under. Port radar, which I go to waypoint 6. See.
I think we have the solar scan over there. Or rotate stuff.
Okay, it just says someone, that can be you.
Super small.
I can do it if you want. I'm gonna be on watch at 3 o'clock. So are you.
No, I'm not. I have meetings all, I have the cruise, post-cruise meeting at 1 and, I mean, Greg and Kira need to stand watch even. BL. Navigation
Right,
hits.
There's a target. So these are the things we're picking up in the sonar on the ROV.
Not a barrel.
We have no idea what that is. Doesn't look like something that might have stored chemicals, although who knows.
It had a lid.
So
It's a
we're
box with a lid.
not
You
gonna.
want us to go more south?
I'm thinking, well, I'm just worried about our radius. Is there a way, Jimbo, to put up where we're
We're-
permitted to dive?
Well, we're that's not a problem. We're not going to get anywhere near our 2-mile limit.
We've
That's
been
not a
heading
problem.
north, and
It's not a-
we're at the north end of it.
I don't
Right,
think-
there was a circle of
yeah.
targets, and we're in the north part of the
Yeah,
circle,
okay.
and we've been driving north. That was my
Okay,
question. Are
okay.
you sure?
No,
that
not
we're
sure. Not 100% sure.
I mean,
Just think
would
it's
you-
unlikely we went that far.
Would you like to look north
We're going
or
south.
south?
We're going south.
We're going south?
Yeah.
Oh, uh-huh. I
We're
mean,
just
south
trying-
is up.
We're just trying to cover everything.
I'm running a grid. I'm not shooting straight where I'm doing this. So if you look right here, I came down, I went up, came down, went up. So right here, I started coming up, got a target, and then going down and up. As soon as we get a target, I go to it, nothing's there, and I turn around.
Yeah.
Yep, head down.
Yeah.
So we're not double
Now I
tracking.
understand.
Can we try to run right over some of those targets
We have.
in there? Okay,
We have
perfect,
been, yeah.
perfect.
So
yeah.
like, what I've been doing, after we left this barrel, we hit these targets, and
Right.
then I started coming up, hit that target, come down, and then as soon as we hit, don't see anything, I'll go to this target. And then we start seeing stuff there, then started coming down, saw something here, here, and then I made a track back this way, started going this way. Just looping around these targets and finding other targets in
Yeah.
between.
Because our sonar reaches
Yep.
out 50 meters.
50 meters.
So,
Wow.
All right, so now
Kind
we're
of running a
heading
grid,
south.
in a sense, but just chasing the targets as we see them.
Okay, so the barrel density here is clearly not super high.
Right in this area, yeah. Although that could change at any time. That's what we don't know.
Nothing there. What is it for?
Needle on a haystack, for sure.
What?
Needle on a haystack, for sure.
It's feeling that way now.
One of the reasons we have so many jellyfish around here is that jellies do really well in low oxygen. They can store oxygen in their jelly, in their mesoglia, and so they seem to be more hypoxia tolerant than a lot of the other animals.
I'm already scanning that area.
But what we aren't finding here are asteroids, brittle stars. Sponges seem to cope pretty well. They just need some hard substrate. So every time we come up on one of our hard targets, it seems to be covered with sponges. We'd say that the jellyfish abundance is pretty high. Sometimes you see them on the bottom. I think they bounce back and forth between the bottom and the water, and they may feed on the bottom, actually. So now we're at a target, one of the Terrell targets, and we're not seeing anything.
Update waypoint bridge.
Somebody's asking what would jellyfish eat on the bottom. Well, there's probably a lot of small crustaceans, amphipods and things swimming around right at the sediment surface. They have stinging tentacles. They can pretty much eat whatever they want. Guessing. Copepods, amphipods, isopods. Not sure though how many invertebrates actually like living here for them to eat.
Where are you drawing?
Siphonophore. We haven't seen too many of those.
Where are you drawing? Where are you drawing?
Hey, Alex, you got us a clear counts? Yes, sir. Tree was out? Is there a permanent tree up? Waypoint 8. No, dude. Follow these down. Third.
Should probably take another two, Dave's highest concentrations had nothing to do with barrels. So, at some point, if we're not seeing another barrel, we should do another two. We have 3 non-barrel sites we can get 2 cores, so we can do another 2 at some point.
No.
Well, I don't know if
We're not going
we
after
can
that.
time it to a slurp for break or something, I don't know what he's interested in, but.
Say the word if there's a place you want to sample.
You guys have a target in mind? Are you seeing anything in the sonar or?
Not yet.
Not yet, okay.
It's a little barren out this direction right now, but we're still moving out. Trying to find yours.
Go back to the starboard, possibly. Hang there. Rotate starboard. Rotate starboard again. Rotate. Alright, hold there. Frame.
I'd like to know how far we are from the Valentine site. Can we ask Jimbo? Do you mind?
We have to, I don't know if Jimbo can calculate that, we might have to ask Deb to do it.
I think he can.
We're
How
pretty
far we are
far.
from what?
The Dave Valentine barrel site.
Jimbo, do you know how far we are from the other site? He's good.
Jimbo doesn't like you to finish your sentence.
I see that.
Not the other dive site, ours.
Right, I've just put a little map up there for you, look that way. The yellow thing is the ROV.
Well, that's pretty much what we're seeing here, Jimbo.
He's asking about a specific site.
Valentine's barrel sites.
Isn't that this?
No, it's at the very, very bottom. There's a circle, a 2-mile radius.
Yeah, but that's this.
Right, and at the very bottom are Dave Valentine sites, and I'm wondering how far we are.
I just see a circle.
You see a circle of targets. These aren't marked on here. To the cluster on the lower part.
Hang on.
Can you tell me how far that is?
It's like around here.
4
Anything, Paul? Nothing, so
Nada, yeah.
even going over these diamond spots over on the map,
Yeah.
There's some little things.
just, yeah, itty bitty things.
Distance 1,490 on the platform.
Okay,
Have a
about to
little
that
thing that's
cluster
directly in
up
front of you,
there.
Betty, tell us where it gets started.
Yeah, or even in a little, just in the middle of it.
Around that.
Yeah.
633.4 meters.
You want us to just start traveling south towards it?
Well, I'm looking at this map, see what, see there's a cluster up there. I don't know that these targets are barrels though. We know the real barrels are at the bottom of that circle. That's way too far for us.
We can just start heading south. See when we start hitting patches of them.
Let's do. Okay.
Brilliant, ROV.
So for people watching, we just had a little consultation about a strategy to try to find the
Oh, we
barrels.
bridge.
We
Yes,
are,
we're gonna be doing quite a long, vessel move down to the southwest.
Can I zoom in on
About,
that?
300 or 400 meters. And we're gonna wanna go at
Where are
decent
we?
speed.
Hold fast.
It's a Kelp hold
6
fast,
possible.
yeah.
Where it is not supposed to.
Yeah, sure, understood. I'll change to
Covered
close
with
speed.
astrus snails.
Roger that. You want me to give you a waypoint or?
Yeah, if you can, that'd be great.
It's gonna be
What?
pretty far out, so I'll give you.
We're just taking a look while we're getting set up
There's
with
an
the
updated
ship.
waypoint for you.
I'm not slowing things down because
Well, I
they
didn't-
have to.
I didn't know if it was just something.
Here, right?
Yep.
Snails, one of the few animals that seems to tolerate the super low oxygen here.
We have some of them along to get all these blue dots everywhere.
Okay, so we are not
Yeah, we
finding
bridge,
barrels
just,
right
bring
here.
that to full, so change heading a bit before the move.
Understood.
Shouldn't affect us.
Who's that from?
That's
Great.
great. I think we should do it.
I can't. I have
You have-
to. I have a ship meeting and.
I can do this by myself too. It's not hard.
Okay.
Don't
Alright.
need two people.
You ready?
There's not a whole lot of data logging going on. And you have an interview at 3.
You know what we're gonna say. You can do it, and I'll sit in here if you want to do it.
Not necessarily. No, cinder bros.
On the square.
I mean, they're pretty uniform. Those whatever they are. There's a lot of them. So what we're doing now is transit over to an area with more targets that appear in a line. We think the ship disposal activity occurs dumping barrels as the ship steams. that would create a line of targets. There's no guarantee that these will be the DDT barrels that we're after, but it's about 400 meters away, the start of this line, so we'll just see what we see on the way there and when we get there. It's a little part of working in the deep sea. It's hard to find things, especially if you're not sure where they are. I will point out that we successfully recovered 4 sets of colonization experiments that were also like a needle in a haystack, but we had a good location information and we're able to land the ROV directly on them in 3 out of the 4 cases, and in the 4th case it took about 20 minutes of repositioning to find them, but we got everything back. So it is possible to navigate down here in deep water, but in the case of these barrels, they were dumped 40, 50 years ago. Nobody has precise location information, and so we are using technology the best we can. The problem is that the barrels sit less than a meter off the bottom in most cases, and they resemble many other hard targets that are out here. There appears to be some military ordnance and other kinds of debris, rocks, that also look like targets. They really don't look any different than a barrel acoustically. So we're having a few issues finding what we're after. So if it looks like we're roaming around a little bit blind, that's what's happening.
Yeah, that's the first time I've seen that.
What? Ship?
ROVs ahead of the ship. Pulling it.
We're not under the ship, are we?
Ahead of it.
Oh, ahead of it. How can that be? I guess we just drive forward, right? it was just me tootling around here without looking for barrels, I'd stop and sample the holdfasts because there were interesting organic falls. And if we suction them, we'd probably get all kinds of interesting animals. You see something ahead?
Possibly one more. Yeah, just forward. Slide forward. Got hold. Alright, got possibility of
Four things?
yeah. So let's look for this one. So I don't know. I think you want to report. What do you think?
Yeah. What I'm gonna do is I'm gonna slide forward.
I'm wondering if we should be doing transects along the bottom.
Not much out here.
But I know. It's a basin. It's an nearly anoxic basin.
Push forward. Push forward now.
This has a smaller, but just like that.
Rotate this way.
Starting to thin out.
Hold it.
We're putting our sonar to work.
A little more stronger.
Rotate. Hold.
We're definitely by some targets.
Should be coming up on it any moment now. There we go. We
Is
got
that it?
and that.
What is it? Can we tell?
What is that?
That's the target?
That one is
That's
the-
a hold fast.
Yeah, and then there's another one just to the right. And the other
Oh.
one, if you rotate starboard,
That's another hold fast. Wow, and your sonar is seeing these? They're about 10 centimeters off the bottom, right? And there's another hold fast. And another. These are kelp holdfasts. This is the attachment site of kelp, is a complex set of almost root-like structures that attach to rocks above the ground. And they often end up washed into deep water. So that's what we've been looking at.
Report back. Hold.
So that was them, huh? Those were the targets.
we gotta have the target
Report
right.
back.
It's not looking big, but
Hold. On the shadow. Report back. Get in. Forward, stone.
so that's a shadow.
I mean, although I would really like to find the barrels so that we can do our sampling, it's a box. It's also good news that not so much of this terrain is covered with barrels, DDT waste barrels, as we thought. So, looking on the bright side, things are maybe less contaminated.
We're almost
Than we
towards
expected.
that line.
So, are we going to that line or this? I can't
I'm going to
actually
this
see.
line
If
right
you look
here.
at our screen.
Oh, there. Yep, I see your screen.
But it could be off of that. We'll search around that area. Till we find our own way.
What's this? Is that an animal? Oh, just a jellyfish. Oh, it had a barrel edge appearance. We are getting a good tour of the basin here. We, Kat is asking about barrels. We do have the hard targets from Jason's dive, and that's probably where we will visit tomorrow. We were trying to explore an area we thought had barrels that had not been visited before, but clearly we're not having a lot of success.
Take that little white thing right there. Here, that little vest.
Another hold fast. They seem to be the target of interest out here.
That in the box.
Lots of
Yeah,
kelp,
boxes.
kelp and boxes. I wonder what those boxes are, though.
Mhm. They're very interesting.
No, port, part. No,
Port.
So
See it?
you have that one, and then.
I'm going after that one over there.
The port one?
Yeah.
Like that.
Are you dropping points on these or not?
I figured we'd do it when we found barrels, but. if you
I'm,
want
do you have a way to change the color of your points or something?
Our points would be green, a little bit more stubborn,
Sure.
No, but I mean, like, can you drop a one color point for things that aren't barrels and another color point for things that are?
I think that might be a possibility.
Well, the markers. Wait.
I'm just thinking that if we can
Sure.
mark all these hold fast, we can compare them to Eric's targets and see.
I've been logging the hold fast.
Oh, you have been?
I have been.
Oh, never mind, then we've got it. Got it on
Yeah,
our
every
record.
time he presses the pitch, it will log everything that's Sebastian has.
Yeah.
Turn right.
Okay, so you've got everyone,
Pitch.
pretty much?
Pretty much, and then I'll put in a kelp fall or a hold fast, and then I'll know
Okay.
that it was a possible
Okay.
waypoint.
There's
target.
another. That's a box. A brick or a box? Brick?
Somebody got in that box. Got another target to your starboard.
Okay, so that was this one here.
Okay, so it's not too far off, but
And then that one was certainly
it's
here.
a little bit more starboard. Yep, right on the flank edge. Right there. There it is right there. Target acquired.
Oh, is
That
this
was-
a barrel?
That was a barrel.
Okay, let's stop and take a look at this. Are we able to?
This is a barrel. Sure.
It was a barrel.
It might have been a collapsed barrel.
See two rings. Look in the set camera over here. See the double ring where the center collapsed out of it.
Yeah. Let me consult with Paul on whether he'd like to sample this or not. Okay. Our trusty science WhatsApp.
Come back to it. We'll come back to that one. We know exactly what's that. Target.
Looks
to
pretty
starboard.
old. Did you drop a dot on that?
I did, yeah, we'll go right back to it. We can check out this target, a little more starboard target. Turn around.
Hold fast. Sort of Murphy's Law. As soon as Paul goes to lunch, he'll find something.
What's that?
Murphy's Law. As soon as Paul walks out of the room,
Do you think you're gonna wanna sample at this one?
we might.
Okay.
I'm not overly excited. It looks awfully old, and I don't see a halo or anything like that.
to.
Can you hang on a second, and let me get the answer?
I'm gonna, if you want to back off just a little bit, I'm gonna bring you down from the high rise. Show you all that. So just find the where the curves go, and then they go perpendicular to us. It should be where you're facing north or south.
We have them all. You saw my message, we're way too far away to get there today, right? It's like 4 miles. But we have them all mapped. You saw them on the chart last night. So we know exactly what they are. We'll go there tomorrow. We'll be better today. I need somebody to run down. Joanna, would you mind running down and just asking Paul, does he want cores here? At this barrels, or does he want us to keep looking for a better barrel? There's no halo. It's really old. We don't know if it was DDT. I know, but he's in charge. So he's at lunch. Yeah. Thanks. Yeah, we do know exactly where we're going. We're on a map. It's- Okay, we've just sat down at a collapsed barrel. Again, we don't know if these are DDT barrels or not. But we will be sampling cores when the water clears. The
Huh? Unless he was gonna let the rest, that's what it has.
Going east to west, going right the other house.
so maybe we can set up. You can orient around it to
We all got started.
wherever you want to be without the stuff blowing at you.
Northeast,
But not for any big cars.
yeah, so it seems really, current's really low right now, that's why it's staying here.
It's probably slack tide.
Barrels out there. You good, we're good to start sampling?
Yeah, we.
All want samples from this one.
What?
All want samples.
Right, we're about to start sampling. You okay to go?
That's great.
Fortunate.
Did you get something to eat?
Yeah. Feel free to go.
So, are you gonna fill this out as well?
Yeah, Chris.
Do it all.
But I don't mind waiting till 12.
Ribs, big salmon, we're really looking kind of good, but didn't have a fruit on my plate. After I put the ribs and the salmon.
What number is he on? Presumably it's 9. Yeah.
It is
I can do that.
Are we on 270, guys?
270, yes.
We are on 270, yes.
And this is gonna be 270D.
Yes.
This is gonna be barrel 2.
Barrel 2. Roger that.
Turn the jar tray on.
So, just, on.
The Valentine barrels that were mapped, did he send you the coordinates? You took them off the Jason?
Yeah, did it. Jason logs. Although he may have sent me something, let me check.
Go with it or let it go?
Is it disturbed on top?
Yeah.
I would let it go, right?
You can quit. Is that number 10?
No. I think it's, well, let's see. I don't know if,
Matter? I can grab whichever one, right?
no, it doesn't matter. What number push core are you on? 9?
9 and 10.
Yeah.
Currently picking up number 10.
Whatever one you want, it's fine.
Yeah, I put 9 back in after Phil. Let it sit for a little bit.
This is 271.
this is 9. It should be 270, right?
I think it's picking up 10.
He's
Okay,
picking up
10
10.
is 271D, correct.
271.
Great.
Barrel 2.
Barrel 2.
Now it's locked.
Your handwriting is really hard to read. What does that say?
Yeah, I'll start printing more kind of so you can read it.
3 meters up.
More corroded than 1.
Yeah, so you're just gonna remember, just come up, just touch, just a little, if you can, just slowly start springing forward. So you're just trying to find it, if you lose it. This one's shorter than that other one, so you might lose it. Avoid it, now we just sometimes. And do you want me to finish it for, or do you want to get in now?
How many more again?
Six one?
Sorry?
6 1?
6 more?
Yeah, you.
Okay. What do you want to do?
It's up to you.
You could have lunch.
He sent you something. That was in October.
Right, so
Yep.
we understand how we're doing this. So you have to run the.
We have, I mean, we pulled those coordinates off JSON log also. I mean, Carlos just sent the coordinates today thinking we didn't know where the barrels were.
Bring it down to 10 meters up here. Right? And high on your resolution. That way you can see really good signal. You don't want to bring things up right around 5 meters from crystal land down.
So you might have to talk Ben through it.
Okay.
Okay.
Sit down, you're taking 2 yards, and then come forward, you just go to right before it disappears, but it's just gonna disappear on right.
I don't see a halo.
And you notice,
So that again,
and you'll lose it,
Yeah.
like, right around here. It's just, unless it's really far, so, like, like, see how he has it right there? He goes forward and he wants it, so that's right at about 3, okay, so, but it's still showing.
Yeah, it's within a couple of meters, they said, of the actual target.
If it's out here, that's straight on. If you line up right here and just go at it, you might actually miss that. You won't see it. So make sure everything that you are searching for.
I mean, it's a barrel, it's just pretty old, collapsed.
Yeah, and it'll be about there.
Contents are gone.
So, when you're out of it. Another thing to.
We should make sure we get topography of the barrel itself.
Way up there.
Did they do that before?
We sat down and there was a cloud of sediment and they started sampling. They sat down 5 meters away. So you can
So-
see the,
Manny, we are doing the second one, so this one in front of us. And that'll be, when you put it in front of the porch, that'll be 2 meters from the barrel. There'll be 2 there. 1, 2, put it away, then reach out halfway. 2 more, and then I'll fly you up to the close next to the barrel.
Okay, this is the barrel. You want 2 months.
You put two right for the stuff. But collect the two first. Put one down, second one down. Then put
Yep.
it away, put it
Gonna
away.
explain to Greg the numbering system.
Oh, there it is right here. So yeah, we'll put it.
Do you want to sit here?
Well, on the spine of my such and two like right here, and two right there.
I'll explain
And then I
to
move
Greg
up.
my system.
Yep.
Just so he knows what we're doing. What we've been doing, you know, the pairs, the zones are
yeah.
called A, B, C, and D.
Yes.
And the pairs are getting written down in order, even though they don't always collect them in this order. You know, we're not waiting till they get back in the quiver to number them. Just like the next pair is gonna be 272 and 273, zone C. We always start at zone D, which is the outer one, and then it's C, and then it's B, and then it's A.
Yes.
Yeah. So that's the only, and I've been,
Is that the actual push core numbers?
what?
You're doing them in order.
We are doing them in order,
Okay.
and make sure we're labeling the barrels as barrel 1, barrel 2, barrel 3.
Yep.
So it's pretty straightforward otherwise.
Okay.
Hello, girl. Ready?
Good afternoon, Chris.
How's your day so far?
There's no push core 11 labeled, so we'll probably have to add it, don't we?
Oh, whoops.
Unfortunately.
I was checking out samples from previous days. Good.
Alright. Leave it in, leave it in, leave it in. I'll leave it here. Yep, leave it in, put the second one in. And then put them both away, that way you don't disrupt you'll get the stuff below. I'm gonna pour it back out for you, hold on. Alright. I ate too much.
The spinach thing? Or the parcel?
I ate everything.
I just had one of the parcels. They're good.
And they look
Mushroom.
good. Yeah. I didn't end up room in my plate for that after I put everything else on my plate. I was hoping that Ben would get one so I could have a bite, but he didn't. So he was very considerate of my needs. You should be able to see this one. Yeah, there. Unfortunately, yeah, relatively.
So just to remind everyone, the sampling strategy here, we're starting about 5 meters away from the barrel, and we've already taken 2 push cores there, and now we're slowly working our way towards the barrel, doing 2 push cores at each of 4 different positions. So the last one will be as close to the barrel as possible. So we're trying to do this with disturbing the sediments as little as possible, which is a bit of a challenge in itself. It's also been pretty difficult to recover these core samples because some of the sediment's been very, very loose and almost gelatinous, and tends to fall out of the core, out of the tube when we lift it. So it takes a lot of finesse on the part of the pilots here to pull these cores up and retain the sediment in the tube and get it into the quiver where there's a stopper in the bottom. And so once they place it down and put a little pressure on it, that seals the stopper into the bottom of the tube. and secures the sample. So this is looking promising right now. Nothing's falling out, and so it's just a matter of getting it in time. And it looks like Ben's gonna succeed with this one. And then you'll notice once he gets it down there, he'll give a little push on the top of that rubber. Looks like a roller skate wheel, and he'll now, there it goes down, and that really seals it in. And so when we get these back up on the ship, we take them apart and we extrude them, pushing from the bottom up. And as the core starts to come out of the top of the tube, we take slices every 2 centimeters and basically dissect the core, which in effect allows us to go back in time as we go down into the sediment. These barrels were deposited, we believe, in the 70s, and there's probably between 4 and 6 centimeters of sediment that's accumulated since these barrels were first dumped here. And so we expect the highest concentrations of any types of chemicals that we might see would be down in a section of the sediment core, probably between 4 and 6 centimeters below the surface. And so we'll section these and we'll analyze all of the sections going down and see where the highest concentrations are of any sorts of DDT related compounds, which is what we're focusing our studies on. But we'll have these sediments available for other types of studies as well, as we learn more about what might have been in these barrels when they were first dumped. So 2 successful cores, so that was what we're calling zone C. So 2 more zones, and so probably going to have to reposition the ROV to get a little closer to get these next 2 samples. Another pen.
Hi everyone, so we're just having a shift change for the science team, and I'm now with Greg Rouse here from Scripps with Paul Jensen, and we'll be here for the rest of the afternoon. And we've got Nico Mojinardino Koch from my lab and Michelle Gurayev from Lisa's lab, who'll be doing the logging for us. This is Sebastian Dive 450. We're at 888 meters between Los Angeles and Catalina Island. And we'll be in this sediment hunting for barrels that were dumped with DDT several decades ago, and doing push cores and taking scoops of sediment and potentially slurping the surfaces of the barrels in the rest of the afternoon. Hi Kyla, hope you're doing well.
Man down. Interesting. Go right between those two. Go right, yeah. Between 1 and 2, or 1 and See all those ordnance-like things? They look like the ends of missiles or something.
Yes.
We're ready to get over there.
There's lots of mysteries down here.
They were talking about one that was from Australia, a paper, a minute ago.
Hi, Dewey. Hope you're doing well. If you read the LA Times story, that gives you a lot of background. So we're just finishing the third pair of cores. Sorry, second pair of cores from here. Then we'll move in a little closer and then be right up against the barrel for the final pair. Great, Ben. Thank you.
Make it?
Yes, both good.
Yeah, we're gonna move right next to it.
Oh, this is B.
275B. Push core 14, barrel 2.
Sorry, I'm confused. 7.5. What happened to... Oh, 7.4 and 7.5. Got it. This was 7.3. 7.4. Okay, so now we're going to be up against the barrel and we'll put the cores right in close. The ROV Sebastian is a large vehicle, and very difficult to land it on such light, fluffy sediment without disturbing it. So it'll clear in a minute. Once we've done the final pair of cores here just adjacent to the barrel, we'll zoom in, have a close look at the barrel, and we may then also slurp the surface of the barrel to get some of the animals living on it, sponges, anemones, other things. We did slurp a rock earlier, which we can consider as really a background animal that's not influenced by the barrels, and then we'll be sampling on some of the actual barrels as well. And any potential DDT residues will be analysed in those animals. Yeah, as close as you can, Ben, to the barrel. Yeah, something like that is good. Thank you.
Sounds good. At least since you have two sets of floors now, really accurate.
Yeah.
Get ahead. Okay. Sounds good?
Yeah, that's perfect.
Okay. I'll ask her. Thank you.
So 15?
Yes, 15 and 16. So one of the things that we're able to see that you're not able to view at home is a map of the bottom and all of the targets that have been detected when one of the script ships came through and deployed an autonomous vehicle to map this area, looking for objects that were extending above the surface. And so we've been taking the ROV and zigzagging our way through those targets that were detected during that survey and trying to get a better sense of what they may actually represent. And in this case, this is clearly a barrel of some sort and is exactly
under one of the targets that were detected in that survey. So that tells us that, well, at least this is, you could view this as a way of ground truthing the data that was collected during that survey. And so for this target at least, we're able to confirm that it is one of these barrels. Just got that one. Montrose 16, 1923. Stop.
And then push it down.
That's 14, right?
Yeah. Or adjustable.
We think this could be a world's record for the number of push cores ever loaded onto an ROV. And that created some complications for the pilots to get it set up this morning and last night. And one of those complications is getting them to fit when the drawer is drawn back in. As you saw, it just hung up a little bit. So hopefully that fixed the problem. We'll see. Nope.
Alright, so it's gonna do it now.
So just leave it slightly. Yeah, leave it there. And they can still fly with the porch out.
What do you want to do? Get some seven.
I should, look at the barrel.
Okay.
This is a little slope it. So the oxygen level is very low here. It's nearly zero actually, so much less than near the surface. And yet we still have sponges. There's a pink scale worm there. There are tube worms. There's lots of little snails here called Alia permadesta that we commonly see in low oxygen environments. So maybe Ben would like a break from push coring and do a little slurping? Sure. Please? Thank you. Thank you very much. That's 278.
Yes.
And this is barrel 2.
Pushing out, I think. That, one more. Alright.
So we expect to find some small animals as well as microbes in the sediment cores. But we're also interested in the animals that are on the barrels that are in the water column, filter feeding. Most of these animals you can see are filter feeders. And we'll also be assessing those with reference to these DDT barrels.
You just want the top layer or
Ben, if you could, the surface of the barrel.
surface of the barrel?
Yes, please. The worms and sponge.
Stood up, you need to. Huh?
No, I think we should, because it's not stretching it too far.
You ready?
Yeah.
Alright, we're in section jar number 1. Is that it?
Number 2.
So we need to
Yes, please.
indent. Alright, number 2. Ready?
Right over here, Greg?
Yes, please, Ben. And up There's a sponge there, so anywhere over this surface would be great. There's a pink worm. Oh. Oh.
Alright, let me turn it off. Read that.
So even in the absence of oxygen, it's still slowly rusted. We don't want chunks of that barrel ending up. It may well clog the hose, which happened to us yesterday. Stopped us sampling a lot. So. Yeah, some of those little shells would be great.
You want to scrape them off?
Yes, please. Yeah. Oh, you got 3 or 4 already. Yeah, that's great. That's good for the shells. And over here, Ben, there's a worm down there.
Where?
Lower right, underneath the. Yeah.
I see you.
Thank you.
Now the small one next to it. Is that? Yeah, it's on.
Yeah.
What? It's not. Yeah, there you go.
These animals living down here must have quite an interesting physiology to survive in such low oxygen.
I can scoot up to her.
Very fragile barrel, so it's quite a challenge to slurp them without them breaking.
Only concern is if I get up.
Just goes to show you that in not much time there'll be no evidence of this at all. No visual evidence that these barrels
Yeah,
were
it's a stretch on that.
dumped out here.
Yeah, camera low, yeah. Alright.
Can you zoom in on this? I can.
This also, I guess, makes a pretty clear point. It would be difficult to try to remove these if there was any thought of remediation. I think these barrels are too deteriorated to think about removing them at this point.
Yeah, but we can.
One thing we don't fully understand is how nature decomposes these compounds, and clearly there's microbial degradation. And it'll be interesting to see if there are certain groups of microbes that are living around these barrels that may be enriched around these barrels and with the capacity to break down molecules, halogenated molecules like DDT.
Yeah, it's just this, the. You know, one more look down. Great.
You got him. Well done.
Goes in, it's flipped to the top. It's very,
Definitely came up. you get one of those little sponges, would be good. It could be maybe the balls, the yellow balls.
The yellow ball sponges.
Yep.
Right down right there.
And there's an anemone there too. Great. Oh, just sorry, Ben. There's that little anemone floating there. You nearly got it. It's on the wide tube.
No, not yet.
Nearly.
There it goes.
You got it. Okay.
Tube and everything.
All right. All we need. Thank you very much.
So Chris, are you gonna head down that?
So we've finished at our second barrel. Today we're hoping to get another 2 more sites. So we'll now move on and look for another one. We've got a lot of different targets to choose from.
So there's a dense band of targets that were detected by sonar ahead of us, and so we're gonna fly the ROV in that direction and see what's there. We're anticipating that there could be quite a few more of these barrels, in which case we may try to find some that are a little less deteriorated than this one, which might give us a better sense of what its contents were. Watch out for 14. Did you bring a lab coat?
I bought a waterproof jacket, which I think will be better. So does Nico. I think the lab coats are going to get just super gross. We have goggles, lots of gloves, waterproof pants,
It's all
sprays
kind-
down.
It's all kind of silly, but gotta do it.
Yes, we will be playing it safe tonight, wearing lots of personal protective equipment, and we've had briefings by the ship's crew, I mean, they don't want their vehicle or their deck contaminated, so there's a bit of a protocol that we're going through to process these samples tonight. The actual concentrations of DDT, though, we expect to be very low. Kelp hold fast.
Oh well, okay. Picture this.
Yeah.
In this low oxygen, things could persist for some time.
Mm-hmm.
Did you drop the range now? Right. Yep, that's it right there. Okay, that's our sonar is slightly off center, so I'll make sure the hand is actually
So well, Greg.
Oh, whoops. We found something.
It looks like spines. That's a whale.
Can you adjust the lights to get a little better view of this?
We'll have a quick look before I get in trouble. Those of you, those of you who know me would know that I would like to stay here now for the rest of the day, but we can't.
Technical difficulties in that.
Oh, something's gone wrong with the ROV. We have to stay here. Yeah. We'll have a quick pan. We could do a mosaic over it. Yeah. Well, first we'll get a sense of how big it is. Let's see if it's. I think at the head of it, it looks like a small whale.
It's only about 3 meters.
3 meters. Yeah.
It's judging from the weight of the line.
Are those sponges? I'm not.
Yeah, there are sponges on the vertebrae, bacterial mat around the vertebrae.
That's so cool.
A lot closer in a second.
Dewey said we have to take some samples.
Unfortunately, Dewey, all we've got is push cores.
In a box.
Yeah. I'm really going to get in trouble if I say anything more.
There's always tomorrow.
There's always tomorrow. Well, we'll ping it. We know it's here.
Is this too old for Osidex?
It could be Osidax on it when we zoom in. It's very low oxygen, but they can tolerate low oxygen.
It's very small.
Oh.
Or it's a ball of something.
Back ball.
Not sure it's a
If
whale.
I may go to my favorite spot.
Give you guys seal. Big head. Big old seal there.
There's some seagrass has accumulated against it. You've got the skull there on the left with a lot of bacterial mat, an orange mat and a whitish mat. And in flying around like this that Chris has just done, we should be able to make a 3D model of this, so at least we'll have that.
Lisa's asking if we should start.
I'd like to maybe do one slurp here.
Right here on the yellow, the orange yellow.
Probably on the bones, but when you're done with the flyover, I'll
Yeah, I did
have
have
a
one around it.
Okay. Can I? You want to?
Would you like to swim?
Let me have a zoom in.
Yeah.
Yeah. Okay. So we've got a lot of the little snails. And the sponges that we've been seeing on the barrels and on the rocks. So this is pretty much background fauna. And we've got a lot of dead sea grass, pyrosomes. The snails are over the vertebrae. The vertebrae are dusted with sediment. It's all animals we would associate with this low oxygen environment rather than whalefall specialists. So I'm not seeing any sign of bone-eating worms or the other creatures we see. The bones are remarkably sedimented compared to other whale falls that I've seen.
I can imagine a story what happened.
Yeah. Okay. Maybe we'll just do one slurp up just behind the skull,
Okay.
please.
We can do that.
Whalebone is full of oil, and that can fuel an ecosystem for some time. Potentially, depending on where the whale fall lies and the oxygen levels and the sedimentation rate, the whale falls can be destroyed pretty quickly, by worms like the Osedax bone-eating worms that dissolve away the bone. Or they can be buried, which does happen nearer to shore, and this skeleton is slowly being buried, it would seem, before it can be consumed. The skull, though, has largely deteriorated. And we're just going to slurp a few of the vertebrae just behind the skull, and then we've got to move on.
What's the bright blue using?
Where are you
The
looking?
bright blue that we went by right next to that big pile of stringy stuff, right in the middle of it, there's like a bright blue spot.
Oh, I see. Yeah.
This orange mat just falls apart.
We could try and slurp and a piece of it might come up. Yeah, we'll try. It could. I'm sure if Ben gently slurps that at the end, that we'll maybe get a piece.
Do a little
Yeah.
metabolomics on it. It'd be interesting.
Great. You want to point out where you want to
Okay, thank you, Ben. So at first you can just slope in around here, and then at the end we'll do a bit of the skull.
We're on 3.
Hi, Eken. Copenhagen or Germany? Yes, that'd be fine, Ben. Ben, anywhere over the bones.
Alright, you're on. Just get a few 4 lights and a little higher. Right, just a bit higher. What was that? Something
We saw exactly the same sponges on the barrel that we sampled earlier, and hi, Bondi. And Mistress. And so the eelgrass there is, we often see a lot of debris will accumulate around the bones of a whale, and the sponges are actually growing on the eelgrass or line. We're not quite sure. Ben's just trying to scrape it off. That's another one, okay. But we're good for the sponges, thanks Ben. You could keep going to the left. Yes, that's great. And I'm just going to look at the bone. See where the... oh yeah. So we have 2 little mollusks here. There's the larger one called Alia. We often see that at the low oxygen zones, but there's a very tiny little glass snail that's called Parvulastrum, and we've seen that on whale falls before. So this is a whale fall worm, whale fall snail that we've seen quite a lot. So if you could slurp just that surface there, please, Ben.
Snails.
Yeah, these little snails, tiny little glassy ones and the bigger ones. Yeah, that's it. And we'll finish with the orange mat, but can you reach back over here?
Yes.
I'm just going to get a close-up of this.
Because we're not
These little snails have quite large feet, and you can see the gills coming out of the snails. Because it's such low oxygen, they need to have a big respiratory surface, so their gills are waving out in the current there, which is quite a nice image. All the white filaments are bacteria, but there are these little snails grazing on it.
Ready?
Yes, please. It's a long name, this thing. It's called PARV, P-A-R-V, A-M-P-L-U-S-T-R-U-M, Parvamplustrum. Okay, that's plenty of those, thanks Ben. And if you can just gently slurp some of the orange, and that'll be the finish, and we'll see if some of that'll hold together in the slurp canister.
Are you worried about getting too much of that in there?
No, that's enough.
Because I would like a lot.
Oh, you'd like a lot of it? Okay.
Okay,
We could, we'll see if it'll hold together.
So this is, the white is likely Veggiatoa, a filamentous bacterium that forms mats and can take advantage of hydrogen sulfide, which it oxidizes for energy. It's usually white, and so this is unusual that this is pigmented. If this is a pigmented form of Veggiatoa, what pigment is it? Maybe a carotenoid, I'm not sure. Or is it another different group of bacteria or something living in association with the Veggiatoa that's creating the orange pigment? So we'll be able to take some of this filamentous material back with us and run some chemical analyses to get a sense of what might be causing this orange pigmentation, and if this is something other than your traditional Veggiatoa. Although I think, Greg, you said you commonly see this orange pigment, yeah. So this is not unusual in that sense.
Wonderful.
What is unusual is to be able to collect large amounts of bacteria. We usually think about bacteria as being unicellular or not organisms that you can visualize. There are really few exceptions in nature. One of them you're looking at here. So these are just large accumulations of bacteria forming mats that you're actually able to collect in bulk and analyze, something that you're not often able to do. One of the other exceptions are certain types of cyanobacteria, photosynthetic bacteria that form mats, and you can collect them in bulk like this. So pretty unusual to be able to make a collection of bacteria in this manner.
Oh, the blue thing? Oh, that's eelgrass.
Seagrass.
I would say some kind of seagrass, and there's a little bit of trash.
That's trash, yeah.
It's probably what we call eelgrass. It's in the coast. It's washed over. Okay, we better go look for a barrel. Thanks, Ben. Nice change from the coring. But we'll do a 3D model of that, and will be interesting to see what's in the slopes tonight.
How long do you think that whale's been there, Greg?
It's fairly sedimented. And with the low oxygen, it could be there for some years, but it's still quite active in terms of microbial activity. So it's a tricky combination of factors to decide the age. But I'd say it would be some years, just looking at the sedimentation on the bones and how buried it is. So there are no obvious ribs. The front part of the skull is buried. There's no big halo of bacteria around the skeleton. We often see that in the first few years, a large zone of bacterial mat all around the whale, and it's now largely restricted to the bones. So I'm saying that that one's not a young whalefowl. But it's small. It was only, I think Ben said 3 meters long, so potentially a calf or some kind of pilot whale. All right, we're heading back to the track now where we got some targets. So we picked up that whale because of the sonar on the ROV, saw it as a target, and it may well have shown up in the previous survey. So what we're doing is, as we're flying along, we've got a map, but we're also looking for sonar targets. And then we're heading over to investigate each one. So our goal today is to get another 2 barrel sites and do push cores and potentially slurp them. And we also need to do some scooping of the sediment to get relatively large samples of the mud around the barrels.
We were going to see there was something there, but yeah, all right. All right.
You guys are going in circles.
Well, we had a sonar hit right in front of us when we came up the whale, so we could go check it out, but there was nothing. So we turned it back around.
So we're coming up to a dense line of targets that were detected by another survey using sonar. And these are basically in a straight line, as if you might expect if things were dumped off the back of a ship. And so we're not sure what these objects are, but we expect we're going to start to see more objects here soon.
Hey Nico, did we log that as a mosaic, the initial flyover? Let's make a note to myself. Okay, thanks.
Moving away anyway. Whatever it is, it's moving away from us.
See how it's circular? Could almost be like the top of a barrel. It's completely buried.
It's black though. I wonder, could it be, there's a trail of it?
Mm-hmm.
Hard to say.
Just keep going.
Yeah, keep going. Thanks.
It looked like it was rock or something, or just net.
It's not. But no obvious, wasn't
Like
power
nothing by it,
signs.
hard. Could it be that the barrel's just completely gone or disintegrated or something?
If they dump the barrels over a long period, some
When was Eric surveying?
March of this year.
March of this year.
Meters away, but
That was big barrel.
What's the distance of your rings in the sonar?
12.5.
Meter? 12.5.
12.5, yeah.
Do you know what makes the big holes?
Oh, I've always wondered.
Really.
I've never seen anything coming out of one.
There's certain fish, I've seen them, they bury their eggs.
Tilefish.
I can't remember.
But it could giant worms. That's what I like to.
Yeah, one pass said 20 meters in the pass, 30. Same exact spot. Yeah, it's nothing. But we're going down towards the so.
Bridge, you have a new position?
Fantastic, thank you very much.
So as we go along this transect, the pilots are using their sonar to detect objects and panning around and looking to see if it's anything of interest. In some cases it's a little hard for us to even see them. If they're the same, if they're covered in sediment, there could be a little bump in the sediment that it's hard to see in the video that they may be detecting with their sonar. A lot of current. It's really the jellies moving along the bottom. There's like a lot of.
Yeah, they're spoken for.
Huh?
They're spoken for.
Dang it. I want to see it so bad. I was going to buy season tickets at feedback because they're coming to feedback again.
We didn't like to get to a single mother before there's no real time.
Or your friend Deb.
Oh, you're my friend? Yeah.
So we're seeing lots of ctenophores. This red jelly you're seeing, very large, it's probably 20 centimeters across, is called Pelagia. And here we've got a
Chris, can we
really
take a look at this spring here, please?
clear circular,
Just slow down and zoom in a little.
got a circular microbial mat coming up. We'll have a close look at that. No sign of any structure associated with it. So it could be something that's buried, but we can't see what it is.
At some point, if we don't see barrels, I'm tempted to sample these, but I don't know if we're at that point yet.
No, I think we'll keep going. Yeah. Okay.
Thank you.
We'll keep going. Thank you.
There's something buried there.
Could be. Here's another one of those Pelagia rufescens. It's a giant red jelly that is supposedly found all over the world. Would be interesting to do some genetics on it in different oceans to see if it really is a cosmopolitan species. It's clearly able to tolerate very low oxygen. We've seen dozens of them today.
So, is it just jelly now? It's no longer jellyfish?
I'd say jelly. No, jellyfish.
And to the left,
Yeah.
where's that white object is.
I mean, yeah, I agree with you. I just didn't know if it was like.
I think jellies covers more things. You just sort of say jelly creature.
Jelly
Yeah.
stuff.
There's we used to say starfish, and now we say sea star.
Still saying
Well, some people say starfish. Yeah. Yeah. There's
We
another
also yellow cats.
microbial mat with no obvious source.
Yellow.
Paul, have you already done one of your cores not on a barrel?
We did one, we're going to do another at some point. I mean, we could do it here, actually.
Would you like to do it here right in the middle of that?
Not in the middle of it. Yeah, let's just do two of the non-Transect cores here.
Okay.
So
You want it right by here or anywhere?
just right here.
Okay. Whoa. I'll need you. I didn't expect that to happen. That's weird enough. Actually, that might have been fortuitous. There's something yellow in there. Now there's something yellow underneath there. Oh well. It's the same thing we were just.
Is that what we were just looking at?
Yeah.
Okay, good.
Core-wise, do you care which one we use?
Nope.
You don't want to continue the number series? Shouldn't it
Yeah,
be-
we can go 17, 18
Yeah, we
is
should.
what you're up to?
Yeah.
But-
You want to core in the middle of that map?
No, I would just core right, I think where we are is fine. No, I don't want to go in the middle of it.
But he could just
Hmm?
get him to use the arm and just poke in there, and we'll just see if there's anything.
If there's
Like,
something
not-
hard
not with the
underneath
crane,
it, yeah, we could do that.
just in case. See if there's any rust or something there, and then it would
Yeah.
indicate the barrel's buried.
Yeah. So it's our intention to do some number of cores that aren't associated with barrels, and this is sort of something intermediate to that. We keep seeing these circular microbial mats, which are a little suspicious, suggesting maybe there's something underneath them that's buried, and so we're gonna do 2 push cores here, and we're not going to do a transect, but we'll just get a sense of if there's something going on here that we can't recognize because the barrels are either completely buried or have deteriorated to the point where we can't really see them any longer.
Yeah, that's good, Ben. Yeah, we'll take 2 cores here away from that bacterial circle, and then Ben will poke at it with his claw, and we'll see if there's anything underneath, as I'm sure everyone in the chat wants us to do. So this is 280?
280, 281.
Lower A and lower B.
No A and B here. Just
2.
no, yeah.
You gonna call it background?
Hmm?
You calling it background? Or
I'm calling these control, no barrel. Starting to lose the tape on some of these.
yes.
And that's breeds challenges.
Luckily they're in order, so we can
Yeah.
reconstruct it on a tape.
Hurry, 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 hurry. Hurry, hurry, hurry. Surface will be okay. Yeah. It's not perfect, but we'll take it. Nicely done, Ben. So these structures that you can see are called push cores. They're a perspex cylinder with a special system of letting water travel through as they're pushed down into the sediment, and then they'll ideally lock the sediment, the mud usually, they hold on pretty well, and can give you a nice section, vertical coring of the sediment that the scientists can then remove that top section tonight and then push the core upwards on a wooden rack so that you can then slice off discs of the mud, and they can do different then stratification of the sediment and then see what happens at different depths through the sediment column. Normally we find the animals in the surface layers. Some burrowing forms can go down quite deep, but the oxygen pretty much disappears after the first few centimeters, so we tend not to find much animal life after that, but there'll be microbes all the way down. And so we're also interested if there'll be variation in the chemistry of the sediment as we go down. So the sediment will be analyzed for at the different depths for the traces of DDT and its breakdown products. And that's why we're sampling here away from barrels to have a reference chemistry and microbiology and animal comparisons for when we sample near the barrels. A couple of the cores are poking up just a little bit high, stopping us retracting the sled. So we'll live with it. And now Ben's going to go over and we'll see if there's anything underneath that microbial mat.
What do you want to do? Greg?
You can just
Steer up?
yeah, see if we can find anything that's generating it. Could be a dead piece of kelp or something else.
Just go down, see if it's soft. If there's something hard underneath it, just push straight down.
Straight down?
Yeah.
Very soft all the way, yucky.
Good.
Yeah,
So,
very soft. It's jiggly.
Very jiggly, no sign of any barrel associated with this
Quite deep.
mat. And very deep.
so it means our controls were good.
Alright, I think that's good.
You gotta clean this up or else it's gonna be smelly.
All right, thank you, Ben. That was a quite a poke.
I poke all the way.
Right there with you, Chris. And you got a Somebody first.
Okay, we're going to continue on looking for barrels. Yeah, it could have been that microbial mat could have been something died and fell there. We were just interested because it was circular. And
So believe it, it's probably sticky.
so there's been some comment earlier today about my t-shirts, and because there's a video of us now posted on the Schmidt website. And today I am wearing an Octonauts t-shirt, one of my favorite programs. Highly educational, even though it's intended for children of 5, 6, 7. It's often backed by really good marine biology, so I thoroughly recommend it. And there are a series of characters. There's a polar bear, the captain. There's a cat, Quasi, the cryptozoologist, and I'm wearing a Quasi t-shirt today. Then there's a penguin, Peso. Inkling is the founder and oceanographer. He's a giant Dumbo octopus. And then there's Shellington, who's a sea otter. And then there's a rabbit engineer called Tweak. And there's a dog, Dashi, the dachshund. And then there are some unusual creatures called Vegemals that are half animal, half vegetable creatures. And every episode is a different marine biological encounter in the deep sea or shallow, so I thoroughly recommend it. And I find wearing Octonaut t-shirts an appropriate place on the Falcor, and rather than at work, although this is a kind of work. So here we've got a siphonophore, and unfortunately we're carrying a load of sediment on the ROV, so it takes a while for that to drop away. So every time we're settling down, we're picking up more and more mud. My favourite character is Shellington. He's the sort of nerdy biologist on the team. But I do like Quasi as well. But then I'm a cat person. But now I'm getting more and more fond of dogs, so maybe I'll have to get a Dashi t-shirt. Nick, who's your favourite Octonaut?
I guess Shellington.
Nico likes Shellington. And your daughter Mia? Who does she like?
She likes Quasi.
Quasi, yeah. Quasi's a bit of a favourite.
That was what we did when we were at the
we're still looking at our targets and traveling along where we would expect to find some barrels based on some previous surveys. And we're looking with the subs sonar as well to see if we can get any targets. There's often a challenge lining up previous survey sites with our own latitude and longitude. There's a lot of challenges in getting these maps to overlay correctly. So it's often what is called an offset that we need to correct for.
Barrels kept disintegrating also. How long has it been?
It's a survey number.
How long has the survey been? Last report. March.
What?
It's about these animals that save underwater research. That's underwater safe creatures. Come on, guys. That's this is like their.
This is
he's
an
wearing an
octonaut.
Octonauts t-shirt. You guys should
This
watch
is
it. It's
classic.
a whole Great
You
Barrier
guys-
Reef movie.
That
on
should
it.
be your evening watches. There's like 80 episodes.
Yeah.
Should be mandatory watching for.
Jane usually picks dolphins. I pick sea turtles. I mean, like, it's a whole fight.
We're shocked. Some of the ROV pilots have not watched the Octonauts
Never.
or heard of them. Oh,
It
wow.
was not. It was
We're-
Cody's wife that brought up the which was your favorite character.
We might have to have a compulsory
Natalie's on.
viewing.
Hello, Natalie.
Yeah. She said she likes it more than the kids do. You're not as cool as Cody's kids, Chris.
It's a cartoon.
Yes.
Yes, you don't have children.
Yeah.
It's a really good cartoon.
Yeah,
What about ocean exploration? You should feature in it. You're like an ROV pilot.
Cartoon
You're
character
like,
drone.
you're like an ROV pilot.
I don't know all the characters, but
But
Not
different.
real
Yeah, like one.
Yeah, they have a lot of different vehicles and they're often in their deep sea suits. And they have an undersea station and little submarines. So yeah, you should check it out. There's always a feature creature that's the focus of each episode. They're only 5, 6, 7 minutes long.
Aw, Carly
Yeah.
said we should make a creature report.
Yeah.
That's the outreach department.
She's listening to our conversation.
Of course she is. She does think that. That's it. We have an LA radio station that was a phone call with these at 7 p.m., which is easier for the ship. Zoom call or satellite phone.
Probably about 80 meters away from the cluster.
Well, we've got a cluster of hits about 80 meters away on the last survey, so we're going to head in that direction. So we've got a dead something on the seafloor, probably a pyrosome tunicate. A lot of these sausage-shaped structures you can see on the seafloor are pyrosomes. If you watched yesterday's dive or watched the Ascent later on today, you'll see them alive. These are colonial tunicates that have probably ventured a little low and gotten caught in too low oxygen and died. In the frame right now are two red ctenophore comb jellies. We're seeing lots of ctenophores today that are able to tolerate this low oxygen level. And the magnificent Paralia red jellyfish that we've been seeing. Yeah, we don't use GPS directly. GPS doesn't work underwater, so we're needing to use the ship's position and then coordinate with the ROV position and then the accuracy of previous surveys that we're using an underwater vehicle, automated underwater vehicle, and it had its own positioning that then had to be coordinated. So you can imagine trying to relate your position 1,000 meters under the surface away from GPS, and it becomes quite a complex challenge. And it's often only a matter of a few meters off, but still hundreds and hundreds of polyps there strung along. So it's a relative of jellyfish, but it's a colony, not an individual. And this one's several meters long, trailing away into the background.
Quite as long as the other one that we found last year, but
No, we need to write that up. Now Chris has got us pretty close. You can start to see the polyps trailing their tentacles. These are probably the feeding tentacles of this siphonophore. There's a kelp frond there lying on the seafloor. Normally they break down pretty quickly, but here we've got very low oxygen, so there's not a lot of animals or even bacteria that can really tolerate this. Here's probably the creature of the day, really, today. It's Paralia rufescens, the magnificent red jellyfish found in the deep seas around the world.
So, yeah, we're just flying around trying to find stuff. We found more stuff that we weren't looking for.
Made my day. Thank you, Chris.
Excuse me.
Well, I mean, you gotta do period.
So our pilots, Chris and Ben, have switched off for a break, and now we're joined by Team Texas. It's J-Rod and Cody. Team Florida's off. Yeah. Team Texas is on. Can we have a look at this, J-Ron?
Sure can. You want to sit down?
Yeah. I can't recognize what that is.
Okay, looks like more like a bowl.
Is it a dead octopus?
Looks like a dump.
A dead Dumbo? What do you think?
Maybe.
Yeah, it looks like it's got flaps, so we're looking at the flaps here, potentially an eye. So
Yeah, I was
we think it's a dead Dumbo. Never seen one of those dead before. We won't we won't poke it. We'll leave it alone. The pilots are trying to. Yeah, let's keep going. Sorry, we can't poke everything today.
to start off. Drop it down.
All right. So the consensus is was a dead vampire squid. Okay. Thanks.
Y'all haven't found any more barrels, huh?
So far, only 2 or 3 really, but
Oh, you found a third
2 were quite broken, and the first one had a hole in it. So
one? I was here for the first one. I didn't see the third one. Still seeing them more? Gonna change my foot pretty hard.
So we're heading to a target that pilots have spotted with their sonar. Vampire squids, well, they're common in, they tolerate low oxygen waters pretty well. They usually found deeper, 1,000 meters or so. They look pretty, and the name suggests some kind of nasty predator. They are actually detritivores, living on marine snow and small creatures. So copepods and other zooplankton that they accumulate. So here we're coming up on a structure. It showed up, and it's a square, a rectangle.
Keep going, right?
Yeah, we'll keep going. Doesn't fit our model of a barrel.
Get a little shadow to your starter.
No, I think it might be just that. Maybe it was a shadow we were testing.
Another little box thing. Circle.
That's a barrel.
Think it's a barrel?
Yeah. Pretty small. Do we have a...
It's like those little prop things that you've seen earlier.
is it more like a paint tin?
Something like that. Really small.
Yeah.
Keep moving on.
Yeah.
Turn back up to the north.
Turn back.
Where you at?
It's a kelp holdfast. So I think I can't see the feed you're getting, but you are getting part of our sonar feed. It's missing a part of the sector, but we're looking for variations in the gold color, and larger objects that are off the bottom will show up as brighter gold color, and otherwise the background is showing up as the relatively even gold specks. And when we see a target, it'll glow. It'll be a lot more intense. It's a big,
Keep going.
yeah, big tarpaulin.
Can we fly right through the little dense patch there? Those targets, just so we
Check mark this.
can,
That showed up as a bright spot on our sonar, but it looked like plastic. We didn't stop to really investigate because we're still trying to hunt down some barrels.
this guy over here.
Not seeing anything in that patch.
We kind of do a 360 right in the middle of that patch.
Pretty much, I'm covering that whole area. Yeah, those the little squares in there are 10 meters. This is reaching out 50.
Just
Mm-hmm. Okay, yeah, it could be an offset.
Okay, you get something coming in now. There's probably one. Or-
I was lateral about 4 when you said 40, so which one, that one, or because there's also one in the starboard. Which one?
There's your starboard. Starboard's a little hard to do. So if you want to rotate starboard, a little more.
Rotate starboard. Alright, I'm holding that heading right there.
Okay, straight ahead. 25 meters.
There it is right there. What is that? Another box. Nothing of interest, Brad?
Not obviously.
could be something rusty.
Does appear to be rusty,
Thousand poop.
but it's pretty square. Keep going. Okay, we'll keep going.
Check it out and then I'll turn far for it. Alright, turning hard for it. You still want me to turn for it, right? Yeah. much stretch do y'all want to go now? We're at like pretty much the end of that line.
Yeah.
Oh,
This is as good as mine. Maybe we just go back up.
we'll go up or
I don't know, what are you feeling?
I'll have to zoom out on that after I check out this target.
25. 20.
So we're feeling a little confused right now in that we expected to see more barrels and we've had trouble finding them. So we're checking out another sonar hit and then we'll maybe reassess our position.
Good. Good to keep going, correct?
That's what we saw.
Just another one.
Another one that's
This whole area right here.
should we just try and take back out that way?
Okay, I can do that. Alright. Start moving the vessel back. Our first one is up right. Way up there.
That right there. The second one is here.
No, they found two. So that the one on the right, I think, was ours or whatever.
I don't know.
Alright, I'll turn. I'm gonna turn northeast. I'll get that. Zoom. Like, show me what. Yeah, it looks like I'm pretty much almost in line with it. Alright, yeah, see, so I'll start looking at that. How's that going? How's that going?
So if you've just joined us or joined us recently, we're at 890 meters and we're on a sediment seafloor at about 890 meters between Los Angeles and Catalina Island in California. It's a very low oxygen environment. We are looking for barrels that were disposed of here some decades ago. And we're interested in sampling near the barrels and away from the barrels to see if there's any residual chemicals associated with the barrels, and then be assessing the chemistry, microbiology and animals associated with that. So far today we've found 2 barrels. A lot of other human disposed debris.
Updated waypoint bridge.
And ideally we'd like to find another 2 barrels today. and we do a series of cores through the sediment and near and far away from the barrel to quantify what's going on. And we're just making a new track, a new path to continue to survey the seafloor. The oxygen level is near zero here, so that's why we're not seeing any fish or any other obvious large animals, except for the things like the giant red jellyfish, Paralia. You can see a couple of them there in the frame that tolerate these low oxygen levels. But we have seen some other small creatures living on the barrels themselves that also tolerate low oxygen. We also were lucky enough to find a whale fall, some kind of cetacean. It wasn't very large, it was maybe 3-4 meters long. And we did a quick survey of that, and you can review back in the YouTube footage to see that if you wish. We just did a quick survey because we really don't have time to spend all day on it, though I would like to have looked at it more carefully. We did make a slurp sample of it, and I'll be studying what that has tonight.
This is what oil and gas looks like.
Oh, really? This is your everyday?
Depends on the job. But like we're doing a freeway survey.
This is what you got.
Mm-hmm.
Making sure there is nothing.
No seeps or other structures.
And then you can target, chase it down, see where it is, just the box.
We'll, the sponges we slurped up dewy, we'll definitely do some chemistry on those and see if there's anything accumulated in the sponge sample from the slurp on the whale fall, and any of the other little creatures we can put through the process as well.
I asked. I even got some sweet red smoke from, or of my coffee in today. I said, does anybody need anything from the galley? Yes, it does.
No, because the galley doesn't have iced coffee.
No,
That's a
the crew
special-
mess, whatever.
That's a special order.
I'm going downstairs. What do you need? That's what I basically said.
Roger that, 4:15.
I'll stop chatting.
I'll stop right there. Straight ahead.
Time.
Go ahead.
That's okay. Just
Start at 5.
Start with five.
Just making that food.
Nobody goes some more. I came down a little bit.
Start with 5.
Start with five.
25 meters out. Straight ahead. Start with 2 maybe. 15. 4.5.
What do you do about it? Hey, do you want a purse?
This.
Anybody want a purse?
It looks like a bear clamp. Or a purse, but let's see if there's a wallet in there.
Or some powdered sugar?
Frankly, it looks like some microbes.
Do you need a purse?
No, thank you. Not today.
Okay.
Maybe it's a sack of money.
It's a sack of something.
Sack of microbes.
A sack of bacteria now.
Ooh.
Probably fell off the ferry to Catalina.
It could have had someone's ID in it.
Do you put chocolate in there?
I didn't this time, but when I made yours a couple days ago, I put a little chocolate milk in it. Just a little bit. My secret.
There'll be a lot of it.
I did just eat a brownie. Dinner. No, this is like the special go in the galley. See, I told you I was going to the galley. Bert's making brownies for dinner.
I see that started.
Yep. It's real.
Turn it hard starboard.
Hard start.
All right, and it is settled down. Now?
Okay.
Was it a nothing?
It's a boost. They say you've seen the ghost by
And we
whatever now.
were quite commonly right just to the left of the 25-meter line. We would occasionally see it.
I'm gonna turn north. Unless you. No, you can leave him coming this way and just delay it. You know, gotcha. All right, facing north. Turn it forward. Alright, I'll stop right there. I'm having change and not up. Small, super small. Okay. Go for it. There. That's one thing, so. Super small.
I got it off the bottom time around 4. So that hour and a half.
Find 2 more barrels, right?
Well, I think if we find one, we'll be in great shape.
those targets that you're seeing there, probably these are a couple of feet away. Come start just a hair. Keep that in front of you.
Hair, starboard.
This starts just passing at 12.5 for a small one. And starboard.
There's something right there, turning starboard. More starboard. Down the rock. Go in this direction anyway. And drain that line. Port or starboard?
You got one deep here and then one underneath here about here. That's true. That's true. This is true.
What do you want?
See, go this way, hit this one, and then down. They're about 10 meters apart.
So I just go straight for it now.
Get go straight for right now. I'll keep these two.
So Greg, this wants to take some scoops at some point before we're done. So I don't know if you have interest in that too. Or where you want to do it.
Yeah, go ahead, Louis.
Start box.
Yeah, did Tony ask
Starboard.
one of the deck guys to open the folding valve this morning? Not that I recall, but I can do that now if you want.
That is.
So that would be fantastic. Thank you very much. Okay, I'll pick up that.
15.
Thank you.
Well, 11. Should be coming out of you. That is. Right box.
It's a something.
Spray something.
It's not a barrel.
So we've seen more of those.
Just
Right there.
somebody dumped a lot of boxes.
Like this time.
What's that thing? This is mound.
So we figure probably need to leave the bottom at 4 o'clock, which leaves us about an hour and 20 minutes left to continue this exploration. We've been running some transects through some areas where we have reports that there's a lot of debris on the bottom. And so far we've seen a lot of debris. We've seen 2 barrels that we've been targeting. Considering how vast this area is that's been used as the dump site and how little of that area we've actually covered, finding 2 barrels is, I would say, pretty significant. But we also understand that there are some hot spots where they could be in higher concentration. It's just a matter of finding them. There's one other area out here that's already been explored with an ROV. That was about 10 years ago, and they found a relatively high density of barrels. And so what we're trying to do now is just get a sense of what's down here, if we can find one of these hot spots. And if not, we may return to that higher density area that's been previously explored tomorrow, for tomorrow's dive. And so our plan is to continue exploring this area that's been mapped but never visualized. And we're probably gonna take a few more sediment samples, maybe using the scoop device on the ROV, so that we can quantify the animals in the sediments in areas where there aren't any barrels. We've seen a lot of these microbial mats, these white areas of bacteria, probably Beggiatoa, suggesting there's some sort of organic source there. We don't know what. We explored one of those mats and thought there might be something underneath it, and poked down with a manipulator arm, but it was just soft mud as far as the arm could reach down, and so there wasn't something immediately underneath it. And so the pilots are very carefully using the sonar on the ROV and identifying objects, and it's pretty remarkable. They're identifying objects that are really maybe just 6 inches or so above the sediment surface, and as they see them, we're heading over and looking at them. And many of these have turned out to be things like that look like cinder blocks. We've seen quite a few objects that are rusty and look old, probably from the era of when these barrels were dumped. They're buried pretty deep in the sediment. Some of those that we've seen have been square, and we don't know why they were dumped or what might have been in them. We're trying to focus a little more on these barrel-type structures because those are the types of containers that might have had chemicals in them. And so we found 2 so far, and hoping we'll find one more before the dive ends. And
2 more.
Yeah, our target was to find 4 today. We had hoped to accomplish that. We really had no idea if we would see any. We've seen 2, which I'm pretty happy about. And so we've got a good set of sediment cores from around those 2, and some other cores away from them as control. And yeah, so it's just a matter of looking and exploring. We're going through this large field of that's been mapped by sonar in the past, where lots of objects have been detected. We've come across a couple of areas where we've seen a lot of objects. Some of them look like they might be military related, looked almost like ordnance of some sort. Some looks recent, like we've seen a blue tarp. And some looks very old, very rusted, and hard to identify, and
No, sir, we're just keep on that way.
quite buried in the sediment.
Trying to find hits.
And so we have no idea what we're not able to see. The sedimentation rate's quite high in this area, and so objects can get buried pretty quickly. Plus it's very soft. If you've watched any of the coring, you can see it's very ooze-like, and so anything heavy dropped over the overboard would immediately sink pretty deep into the sediment, and then with a high sedimentation rate would get buried relatively quickly. But the barrels that we've seen have been, at least half of them have been exposed, and so we're hoping to be able to find another one. We can see, I believe, about 50 meters ahead with the sonar. 50-meter range with the sonar?
Yes.
Yeah, about 50 meters ahead, and we're scanning back and forth, going up and down, and going from object to object as we see them. So fortunately there aren't a lot of rocks out here which would interfere with this operation. And yeah, fingers crossed, maybe we find one more. Otherwise, we won't be pulling the all-nighter we were expecting to be pulling, processing all these samples tonight. So here you can see an object that they've detected and come in to take a look at. It's not clear what this is. If this is, oh yeah, could be a piece of seaweed. It's what this looks like. Kelp holdfast maybe. Seen quite a few kelp holdfasts so far. And as Greg mentioned earlier, since the oxygen's so low here, these don't degrade. Yeah, of course. Yeah. Slurp. Slurp away. So we're gonna collect the sample here using the slurp mechanism on the ROV. And that allows Lisa and her team to examine the fauna, the animals that live in association with these little islands on the bottom of the sea.
I think that in the climate change context, what people want us to see algae as a way to capture CO2, put it in the deep sea, so knowing what eats that, there are different conditions as well.
This when we asked the new name,
Alia. It's great. I know those names, but for like me, I forget them all the time. Did I ever show you my picture of Alia? You're a sailing.
Yes.
It's a clear place filled with nothing but water.
yeah, they have this big foot.
Just put it up like that. But I think it was San Pedro Basin, which I feel like that's where we are. We had a dive here on the Nautilus in 2015, and we encountered all this same stuff, like dead kelp but no barrels. But I don't know. Maybe we were further south. Anyway, but at that time, the water was full of shiny objects that turned out to be Alia. And you haven't seen any shiny objects flying by, have you?
No.
Could be
No, we would've seen.
snails. I think the water had gone anoxic rather than having half of the micromolar.
Pretty low. Yeah.
It is pretty low.
It's got lots of the alia on it.
So, did you have a chance to zoom in on it?
More than this? You can zoom
Yeah.
in? Yeah.
Can we
Do you want
zoom in all
to
the
zoom
way
in
in?
quick? I haven't
There's
used it,
crest
so.
there, too. Get too far.
Oops, sorry.
There's serpulates on them,
Yeah.
so it sort of looks like the whole pass will hang around for a while. Anything else?
Serpulates, alia.
Little white dots are forams?
I think so, yeah.
There's arborescent forams.
Arborescent forams.
What's the white tentacle? Well, maybe a spionid or something.
Could be.
Good with the zoom?
Yeah.
Ready to slurp.
Just anything or?
All of it?
All of it.
Well,
All room.
he doesn't have to collect all of it, just all around.
Start over here. Oh, these worms.
Oh, you can get the worms for me. Thank you. It's lots of serpulates. I'm surprised.
Well, it suggests that the kelp's been down there a long time without being degraded.
Yeah. Well, they can grow very fast, but
oxygen. The other thing is, what, now, how are they breathing? Maybe I'll make my little climate change comment about algae in the deep sea. One of the reasons for sampling this is to try to understand what eats macroalgae that sinks into the deep sea. And one of the reasons for wanting to know that is that there are many different forms of climate intervention to remove carbon dioxide from the atmosphere. One of the proposals is to have massive seaweed farms over many hundreds of square kilometers and then grow algae that sucks up CO2 through photosynthesis and sink that algae into the deep ocean where that carbon remains relatively immobile, and that would be a form of carbon sequestration. And one of the big questions is whether the algae sunk into the deep ocean would actually stay there undegraded, or whether animals would eat it. And I think what we're seeing here is that at really, really low oxygen levels, and it's about, under 1 micromolar, that the algae, at least the holdfasts of Macrocystis, seem to hang out for a while and perhaps not be eaten very quickly. It's hard to know how old this feature is, but there are clearly a lot of animals that are interested in it and probably eating it. Why is oxygen so low here? Well, I think we're probably in a basin, but we're also at depths that are firmly in the Eastern Pacific oxygen minimum zone. And that minimum zone is the product of a very high productivity that results from upwelling, winds blowing along our coast and through the Coriolis force, causing a upwelling of water that brings nutrients, fuels high productivity in the California current system and along the California coast. That productivity sinks and decays. It's degraded by microbes, which consume oxygen in the process. And that water at depths of 400 to 800 or 1,000 meters is intermediate water that's not really replaced. The oxygen's not replaced very quickly. It's pretty isolated. So the oxygen continues to decline. Climate change has made it a little bit worse, we think, but there's naturally very, very low oxygen here. And when we have a basin with sills without flushing, that oxygen can go down pretty much close to zero. I postponed it for tomorrow because I thought we were talking about. No, but I want to make it sound like there's fewer out here than we expected based on the technology we were using, and I think that's a good thing. It also helps us, it's one of the few ways ground truthing that Earth has. So I think it's very important. I would, I'm hoping he's watching. He doesn't see this map though. the whale, your baby whale?
Yep.
Do you have any idea what that was? What
No.
kind of mammal could be that dimension? And
I'll ask a whale in person. It was small. It was only 3-4 meters.
so it could be a young one.
Yeah.
A calf. Yeah.
Or a minky or
Deb's comment was when he filmed the dead Dumbo. I was like, too many dead things down here. This is a feature of oxygen minimum zones, though. Really low anoxic basins.
Mhm.
You got a visual coming up?
Would you consider a lower barrel?
I don't think that's a barrel.
That's not a.
What do you think?
I don't think it's a barrel.
Then let's keep going.
I'll have to look at it from the top with the sit before we take off.
That could be a piece of barrel. But
Looking at sit, it just looks like two wires, really. You see it right here?
Yeah.
we don't know what 4MCs are that are like speckling the entire.
when you lock the barrel on the top.
Yeah.
There'll be those balls. I mean, we'll get some.
Oh,
of
you
this
think the
later.
pelagic ones?
Yeah, I
will
think
come
so.
down rather
Yeah.
than global bulaminas or. So can we? So I'm just thinking a scoop would be good to get some background
Just anywhere?
animals for control
The purple? The
anywhere.
Okay. What do you want? The top 10 centimeters? 5 centimeters?
5, yeah.
Just keep going
Do
at
you
5.
think there's any animals here? I mean,
I'll be surprised. I think there'll be a few polychaetes in the sediment, but not many.
could also, I mean, are you interested in the sponge? We could scoop sponge.
We've gathered the sponge already when we sloped. And we got the same sponge on the whale.
And you got some. That's good.
Yeah.
You didn't see Oceanography?
No, it was covered with sediment.
Is that sponge?
Matt.
Matt. Yeah, I'm not gonna have time to go sorting all the fauna, but we could, I don't know. You know, I was originally thinking we'd scoop and get bigger animals, but I don't think there's any big animals in here.
There were some big things in the push course.
There's some jellyfish.
Big,
Some jellyfish.
there was a big worm in one push core and a tube
that's
came
true.
out of another one.
That's true. If we could get worms like that, was it I think it could have been an area or something. Yeah, if we could get big ones like that, then it would be worth it. I just know Carlos to run a DDT sample needs a pretty large volume of tissue. So where are we going in?
Everywhere.
Worst case scenario is we don't graduate. And it's good for us to test out the technology. All right, I'll consult with Greg. What feature allows us the greatest opportunity to find animals in a scoop sample?
It is holding right
Well,
there.
presumably the sponge is on something a little bit hard, like, so we want mud really. This looks, there's patches of
Lighter brown.
mudstone or something here.
So many little houses on the road.
You want me to, I don't know when you wanna
Do a scoop?
take it, yeah.
I don't know. choosing one place. It's tricky, isn't it?
I mean, you can wait till something interesting shows up, but then I don't know if it's like background scoop.
Yes.
It's background fauna.
Well, I thought when we found a barrel, we're going to scoot near a barrel and then kind of at the distance of the cores away from the barrel. 5 meters, or what have you chosen, 3 meters?
I mean, that was the original intent, to scoop in the vicinity,
Yeah.
but I'm not confident we're gonna see another.
Today.
Sit back down.
Hold fast.
Back to the drawing board, bud.
I think our headlines will go from 25,000 possible barrels to 25,000
Square blocks. Something square.
So anyway, you can take a scoop when the urge strikes.
In the next hour.
Yeah.
Yep. Well, I hope we're going to find another barrel.
Nothing here for you, buddy.
Do you know what slurp number that was that we just took for the kelp?
That was from
Number 4.
Crystal River. You did. Thought you would. Oh, wow. Rude. That's my boy, believe you.
Are you looking at artifacts, Tyler? Different barrels? Maybe even heard, but I'm processing Microsoft system. I don't know if it's not like the resolutions.
Come on, Barrel, show your face.
I can try, but we're not. So tomorrow is a good day. Oh, did you forward me that email?
Something funny?
Got it. It's just that we're on here.
Come on, be a barrel. Come on, barrel. There you got this all.
We're Sonar's picking up the pass. We're not missing one.
No, there was a barrel that we
Yeah.
were. that-
So we're continuing our hunt for barrels that were dumped here in the, well, decades ago really, looking close to 50 years ago. And that were with DDT disposed of between Los Angeles and Catalina Island. We're at 886 meters. We're using our sonar on the remote operated vehicle Sebastian to find targets, and the pilots are actively hunting down these little hard structures. And there's been rocks and boxes and many different things, but 2 barrels only so far today.
That's not a pink sock, is it?
No, it's a pyrosome. Unfortunately. What's our full range there on the sonar, J-Rod?
Right now, or
What have you got what range have you got set on it at the moment?
currently Cody has set at 20, but we've been flying around with 50
Right. So it's 20 to the full extent.
right now.
Yep.
But
So the
he's just showing possible.
right. If you're looking at the sonar on your screen feed now, at the range of it, at the way it's set at various ranges. It's right now on 20 meters. To the so each ring would be about 6 meters. But we can switch it to different ranges. So a lot of the flying around has been done at 50-meter range. We're on 50 now?
So I had put in the degrees minutes seconds.
I'm gonna turn just to the southeast a little bit more, just to see if there's anything out there. I'm gonna turn to the southeast a little bit. I'll wait for the bus to catch up. That's how I know it works. Alright, go forward. Yeah, it's they can definitely see it.
We're just looking at Carlos's descent barrel coordinates, and I was trying to see if they match. They are quite, we think there's a Google Earth map. They're in the vicinity of the Jason Hall.
So are we on 50 meters range now?
Yep.
Yep.
Meters.
Yeah, we're on 50 meters now.
This one I imported, so it would have been a similar area. Yeah. I don't know what David sent on these points, and what I'm getting from him.
So David sent me.
That's what these are.
I would believe David.
That's what these were blue, and
survey done of this area to look for the barrels?
Well, there was the Sonar AUV survey early this year.
Oh, this year.
That's what all those targets are.
Okay, I didn't know if that was from 10 years ago.
No.
That area right there. Rotating corner. A little bit.
That's a lot. 10 times 20 minutes.
Just a few degrees, nothing there to come back down.
So there's two in here, he just sent us that, addition and these
Position, you could ignore those. But those are times on the recent thought.
Yeah, right here.
6, 7, 8, 0.
Right, turn back to my starboard. You saw something up
It's
there.
actually, it's the barrels with.
Yeah, that's close to my name.
But I trust David's numbers more than I'm going to do something.
Yeah, I just,
If there was, like, if we have one where David's and Carlos is converged, that would be a good.
Salt. Salt, pepper. Salt, pepper. Barbecue.
Can we each present one there, or you'll see, it takes 2 to 3. It takes ideally 3 of us.
That's fine.
2 people to do it.
It's like really easy.
And it takes a little while. Get faster.
Starboard? Run that way, starboard.
So, when we have 10 hours, it's not still 20 minutes of work. So,
Oh, it
the
almost
amount
done.
is still
That's
like
me.
And, yeah, so just roll up now, we can do it.
That's why I was asking.
Sure, yeah.
So, David's got a big control.
All that. Y'all wanna do a survey of the whole town?
Yeah, it doesn't,
Right here.
it doesn't update the website until after we die, because the data that's being logged right now is transferred to the place.
So if it's not on vacation,
Wrong.
we can access the.
Right, where should I go now?
When you can email them, they can all file if you want later. Make it.
You don't have access yet.
Good, right?
scoop
rotate
maybe?
starboard,
Soon.
another rotate starboard.
Let's hope we can get a barrel.
Even if we find a barrel at this point, I don't think there's time to sample.
Be a big
It takes an hour.
That's one. Or the big box.
Well, if this is a barrel, then we could get it done.
Buddy behind it. Found another box. Man, I got excited about that one.
Yeah. How big is that, J-Ron?
It's not that big, but it gave us a really good target from our ways out.
Yeah.
We got a bit excited about it. It failed us. Is that a bone? Alright, turn it back to my report. We'll go this way.
All right, we're still looking among our samples, or targets, rather. And there are things down here, but no other barrels since before midday today. In Pacific Standard Time. We are in California, just offshore from Los Angeles.
Not even small target talking.
So Lisa's leaning towards not going back to the sea.
Started pretty hard.
You gonna be pretty upset with
About
that?
20 degrees.
We'll try and get.
No,
We'll try and go there anyway, some other way.
Put it in front.
Put in a proposal.
Yep.
Just-
It is here.
So we'll go to Coronado.
Hmm?
Coronado.
Sounds like she wants to go for that. You don't have any phosphorates, yeah, that we're going to be finding over.
I've always wanted to look at Coronado.
Right down the box. Did they dump them in square boxes?
Not as far as we know, but like jerry cans, maybe. Should we go and poke one?
I wonder if any people alive who were involved with it could tell the story of what they did.
Yeah, could be. Company records.
So if we're off bottom at 4, I'm gonna ask Lisa if she wants to have a science meeting then, so we can get it out of the way. Instead of at 8. Yeah, you good with that?
Mm-hmm.
I don't know if he's got 33 twice, if they're really,
We're seeing these.
We are.
are.
So what?
They're not there.
Ask him to send me his positions, he doesn't produce it. When I take that,
Carlos probably can't convert it, he'll just go on to a version on the.
They were sending blood.
so, but with the numbers I'm getting, I just went through and converted them all. When I do that, it puts them here in the middle of nowhere. So don't know, like they look like they're right in reference to each other, but I just did an entire spreadsheet of like converting. I just don't know whether his.
But, okay, I don't wanna bother,
Okay,
there's a
that's
lot of
fine.
chatter from Eric Carroll and Peggy Batchelor.
nothing.
Yeah.
Sorry, we had the microphone off. We have been talking, but had the microphone off. Pardon me. Yeah, we're going to be off bottom in about 30 minutes. We probably don't have enough time now to sample a barrel. If we find one, it takes about an hour to do the push cores we need. We do need to do a sediment scoop though, so we'll settle down fairly soon and scoop up some of the sediment. And we anticipate we will be back here tomorrow. We're going to reassess our data points and make a new plan, because we explored pretty well what we were given. And so we're just going to check the various coordinates. So, J-Rod, I think we'll settle down and do a sediment scoop unless you've got a hit. Okay.
We are going.
Okay.
Target?
These guys are into it. They're having fun with it.
Well, that's the other thing I think Eric needs to know. We have this sonar down here. That if we were within 50 meters of a barrel, we'd be seeing it.
I mean, we're detecting kelp fabs. That's, they're somewhere, so we're not missing anything.
Right.
No way we're missing anything. Yeah, he needs to know that.
It's not like we're navigating blind and we're just, I feel
It's
like
not
we
like
have
we're-
a better look at the bottom than they do, so.
And it's not like tunnel vision either. Their radar is like this, and we're seeing a fairly wide swath of what's ahead of us.
The first one. One box here.
The box.
Another.
So maybe they're,
And
the
then
boxes is how they put out
I
the
got
DDT.
another box over here.
Those are ammo boxes.
There's a lot of them here.
And some bolt pads.
We used to get those.
There
From
was
the movie store.
there was something right there. And then I had another, whatever it is.
Should we lift one of these and look, see what it is?
Cody said it's what people make, other people go swimming.
Hey, you could slurp that fantastic worm there.
Which one? The one that's
Is
swimming
that a
around?
worm? That's a worm, isn't it?
Is that a worm or a jelly?
Is it a jelly?
It's
Oh, it's a jelly.
Never mind.
Worms on the brain.
It's a jelly.
Well, since we're down here,
Right, got
um,
worms on the brain?
yeah,
Scoop.
It looks like
we
a
should
contact
we
sensor.
should do our scoop.
Like the color
It
of
does,
the
yeah.
It's good.
I thought it was a. Yes, please.
Around a box?
Uh, well,
Just anywhere?
just-
I mean, there's animals here. Look at all the burrow holes. There's somebody in the ground.
just anywhere you want, J-Rod.
Yeah, alright, I am going to.
I mean, if you want to get near the ammo with the box, we could.
I don't.
You don't
yeah,
wanna-
I don't care about the
You don't
box.
care about the box? Okay.
Do those look like ammo boxes to you? Like the Army Navy's war ammo
Yeah, they could,
boxes?
yeah.
Could be. They're a little wide. It's also a little more narrow.
It could be the 21.
Could be a jelly.
Lisa, here's your position on Charles.
Alright, I'm gonna sit down right over here. That's okay. Any issues with this area?
Looks good.
Roger that. Take it down.
So we have a couple of metal rings with a handle that have a canvas bag attached to them. So we're going to drag those through the upper sediment and fill them up, and that gives us a pretty big volume of sediment to look through relative to a push core, which is a very narrow cylinder. And this will give us an idea of much more of the diversity of animals here in the sediment.
Do you know, like, scale-wise, what this would be? Distance? Here? Just from this end to this end, this cluster. Some of those are the ones he said how big those would be.
Not too deep, please, Cody. Just the top
Yes, sir.
inch or so.
What do you do that? Are you gonna open up the power line? Is that what we're on? Confused. Is that what's the a little bit more?
That's a great option. Start over here. You start somewhere right you land on. So you've got one here, and then you've got one here. So you start here, and work your way this way, guys. You can make a waypoint here.
We didn't do this on the Nautilus.
Oh, they're great.
Yeah, perfect.
There's probably reachable on there. You can make a waypoint and just drag, probably find arrows right in there.
It's very fluffy sediment.
This way, this whole thing, like, you go all the way from there to there.
Chunks.
Oh, there's chunky bits? Uh-huh. Maybe they're phosphorites. Okay, it's gonna have to...
Is that the long bag or the short bag?
Don't know. It's bag number 2.
Can you tell if that's our canvas bag number or yours?
Oh, yeah, because it has Cooper's writing.
Does that have a number on it, J-Rod?
There's 2 on the handle.
Number 2.
Do you know if our long bag got fit in?
We didn't have the time last night.
Yes, Dad, I'm ignoring Facebook.
It's always nice to have a parent who's
What's happening? Between 3 and the SB1, right?
Sounds good. SB1?
SB1.
Yeah.
This is
I don't know where.
283.
283, correct.
Very original, friendly. It's,
I thought it was just like an anonymous name.
no. Yeah.
Yeah, so I think tomorrow I just need to.
Do this all day tomorrow and not
Box
see G?
G?
We got lucky to see 2. So that would be not good.
And the two we saw are very old and not active. Like
Are you
these have
doing
halos
one more?
around them.
Yeah.
Don't-
We
There
might.
was nothing.
Do you want the other scoop since we're here? It's a lot of
I,
sieving.
I don't know.
May as well. We can always
Sure,
throw it away.
You can always throw it away.
Yeah, we'll do the other one. Thank you.
You want to move
Yeah.
or?
We could hop forward a little bit.
Yeah, I guess the worst case scenario would clean them out.
You have a 500 micron. 500 micron sieve?
I have only 300 with me.
I
I have
have
a
it.
big one though, so you
Oh,
can
okay.
sieve a lot
Yeah,
fast.
great.
12 inch. I mean, how much is it? More than
It's like a
12 inch.
16 or something. Yeah.
One of, big ones.
Yeah, it looks good.
I haven't clicked on them yet.
You can show him what we were just talking about.
From there,
Who are you
Fox
gonna-
Mill Wildlife.
So, you wanna take a look?
So this is a nice start at the eastern edge of our work area and then work the ROV toward distance towards a minimum of continued territory. That way you get some bearings and find barrel and you can move into uncharted territory. Of course, when you get out to the resort
Okay, so this is the east side.
Take a look.
This one first. And that's the easternmost one that he said, so we're gonna tell him we're gonna start at your BBL 40. Work towards 31. Working west of 31. And then maybe.
And there's some places in between, well, I
No,
guess
the
they
red
probably
is
didn't
like,
find
I don't know what
anything.
the red is.
Can you read the coordinates for me? You start that with you.
33.566. 566.
33.566.
So we'll take this sediment and run it through a 300 micron sieve, so that's 0.3 of a millimeter tonight, and that's quite a large volume. Hopefully, at this sort of depth with low oxygen, we don't expect a lot of biomass, so we'll be looking for small annelids, maybe some mollusks and some small crustaceans in the sediment.
2013.
And we'll probably do a second one, just in case we have time to sieve that second bag as well.
Alright, so we're picking up and moving forward,
Yeah, just
not too far.
a little bit forward. Yeah.
Good to go. Should we type it in?
Okay.
Huh?
So it's not going right of this? Someone's there.
Arlo might want some of those sediments for his embryo assays, I'm asking him now. So before we put it all through a sieve, we might scoop out a little bag.
So I guess Eric classified 20% of his hits as barrel-like. So we've just looked at hundreds
So
of
there
his
should be a
hits
concentration of
and
things here.
2% split.
See where their targets fall on the sentry hits. So there should be some stuff.
I mean, so we could go like this and that. If we really
So we're going to do another scoop here,
The thing is,
and that'll probably be our last thing for today. We might take a water sample. We did 18 push cores today, so that's quite a lot of processing, as well as 4 slopes of rocks and whale fall and kelp.
Almost did. You can't both me and color. Butterfly.
Hmm. Well, we did rig up, had the pilots rig up 39 push cores, so we had big ambitions.
I know you were. It's okay.
We were planning 4 different barrel samples, but we only did 2, so maybe tomorrow we'll get 4. And then we'll use pretty much all of the push cores.
Good Lord.
The whale fall was very interesting.
It's DDRL.
That's the most helpful I think today.
Okay, good.
Okay, thank you. I think those are polychaete tubes called
Wait for it
catopterids.
until I start recording it again, because yesterday I got really feel about that.
Wiggle, 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 wiggle. Shake it out, buddy. Shake it out. That's what gives a good smelling mud.
Thank you. Yeah.
Anybody want to do some facials later on the night?
Now that,
Could be good for you.
So is this a second sample?
Yeah.
sure.
We moved a little bit.
Bag. What was the first bag?
At number 1 on the handle.
Number 1?
No, number 2 on the handle. Sorry, it was the first one.
And this
First
one,
says number 1.
this one has number 1 on the handle, but number 3 on the bag.
Correct.
Got it.
We're just trying to keep going.
Keep us on our toes.
You can't see what the bag says when it's stowed.
Yeah.
But I already made up the map.
You want another pass of that same area?
No, I think that looks good.
You happy with that one?
Yes, please. I'm interested in the tubes you found.
Awesome.
Alright. That's it for sampling, yeah?
Any Niskins?
No. I asked her already.
Okay.
She said no. I think she said no. No more buskin in Niskin. Done with Niskin.
Can we close that for you?
Well, the good news is we don't have to re-prep all those push cores tonight. Or at least not as many as we thought.
Silver linings are good.
You could do anything on your login, or can I leave this up? You don't need to sit there. You need to know. No, okay. Because it's this program running there.
We're doing the baiting straight away.
She hasn't responded, it makes sense to me. She's not answering. She's ignoring my message.
Day. All right, everybody. We are finished for today with sampling, and the pilots are now prepping the ROV to come up.
I'm not kidding you.
We'll be on deck in about 40 minutes or so, and then we'll unload all of the samples in preparation for working on them through the evening. Thanks for joining us today. We hope you'll join us tomorrow, and we'll be surveying similar area nearby, again looking for barrels on the seafloor and sampling near and away from those barrels. And thanks for joining us, and I think they'll be streaming the Ascent. And keep your eye out for fascinating midwater pelagic creatures.
Nice job, guys. I feel like you like drawing that. Like Easter egg hunt or something.
It is. It's kind of fun.
Yeah.
It's kind of
See,
fun.
you're into it.
We like the challenge. Team Texas says you're welcome.
showing you guys the view from our ROV winch camera, and we're looking at Catalina Island there off our port. And if we spin around, you can see coast of California and some rust.
That one's strong too. Cool.
Group getting set up on the aft deck, ready for the vehicle to come back on board.
Oh, y'all got double hoses?
Yup, we doubled up on a.
Wow.
So we're just doing a bit of a up and down on the water column to blow off some of the extra sediment on the skids and the vehicle. Make sure we don't bring it on board.
So for those who may not have watched one of these recoveries before, you have three views on your screen right now. The left is the 4K science camera. The top right is the sit camera, so that's the camera that the pilots fly off of typically. I mean, they use all of them, but that is what is in front of them. And then the aft deck, you're seeing the A-frame with the docking head attached, so, and the umbilical cable. So our Sebastian is an ROV, a remotely operated vehicle. It is completely and always tethered to the ship. It's how we get our video up real time, and how the vehicle has power all the time. So when we recover the vehicle, the pilot flies the vehicle up while the winch is paid in, and then they fly aft towards the vessel's stern, and then when the vehicle is right underneath the docking head, the winch pulls it out of the water. It then goes up into that docking head using the tension from the wire of the winch wire, and it does lock into that docking head with some latches, as then the A-frame is paid in, so it keeps the vehicle from potentially at all coming down out of the docking head.
No, that was a wheel driver.
That was a pile driver?
Yeah. I thought that was a DDT. Was the finishing move of Jake the Snake, where you help them in a headlock, right?
Yeah, I'm getting my headlock straight.
Dramatic.
We will have another dive tomorrow here at the same area. We'll be off deck at 8 a.m. Pacific time.
No, thank you. I was saying, Cody, nice job for even pulling out Dream Team.
Yeah, it doesn't matter if you know the name, if you don't actually know.
And then we will have one more dive the day after tomorrow. Location unknown at this point. I don't think the science team's confirmed exactly where they want to go. Some potential to go back to a previous site or to go to a new site.
We're going to try a couple utils.
And then we will be in port on the 6th. And the next expedition will start on the 12th.
Y'all could not take him by yourself.
I believe that.
I don't even know if both y'all could take him.
I'll take him. I'm not that dumb. I mean, if I was going to try, I'd break a stick.
Oh no, he has been working on his dad bod.
The plan tomorrow is to set down on some of the barrels that they surveyed in 2011 using, in 2013 using Jason.
Oh yeah? Big car carriers? Typically, what ships do. I like to float. You boys, that ship out there, just that one in
The
particular?
next expedition will be off here, the same sort of location off of the coast of California. We'll probably be out in the deeper area off San Juan Seamount again. It'll be a complete midwater expedition, I think with maybe the exception of one dive. They are doing a number of different, it's a bit more of an engineering but also midwater collection, so they have a number of instrumentation to scan midwater organisms as well as collect them. So it's a group from the University of Rhode Island and a group from Mbari. We did a one-week engineering cruise with them off of Hawaii a couple years ago. This cruise was supposed to happen a couple years ago, and because of COVID has been delayed, so we're finally getting a chance to have this team on board. So Dr. Brennan Phillips is the chief scientist.
Hey, tell me if this works. I can only take you two jackasses talking so much on the radio. I have to correct at least every 10th thing you're wrong about.
I don't believe the focus of the next cruise is fish, so I believe it's more gelatinous organisms. But I'm not entirely sure. I'm sure that the science team will go over that in detail when they're here. How much pressure can Sebastian withstand? So Sebastian is rated to go to 4,500 meters. I'm not sure off the top of my head what that is in pressure, but that's the deepest we can go with her.
Depends on if Sebastian's gonna snow when he gets up. I mean, it might have a little steep.
I've been rinsing it off pretty good, getting big clumps of mud going on. But there are the pork shoots. Now some pretty warmy mud. I don't even know if that how you call it.
No, Nudie, I don't believe that he will be involved.
Cars that y'all take out of that, one in front of the biobox.
What?
Do you know what cars was taken out of the one in front of the biobox?
The first two of that group. So
14 and 18.
33 and 34.
Oh.
Or 34, 36, yeah, no, 33, 34.
So the dives on the next expedition will be a couple of different times, but typically they're planned for noon to midnight Pacific time. But there may be a few days where they're slightly earlier. Stay tuned.
That's the only form of communication you understand.
It's not true at all.
You know it's kind of true.
No, it's not true. Hey, and I did appreciate Facebook coming back and backing up my side of the story. I didn't have anything to do with that Jeep picture, but we do have the Jeep picture to show you. What? That Jeep picture. Of the somebody posted it on Facebook when they heard we were working together.
So if you have a good eye in YouTube, if you look in the A-frame camera, you'll see there are little yellow dots way off in the distance. Those are our floats, just got on surface.
They fit on this hard hat very well.
Yeah, but they don't get close to your ear because they're designed for the baseball caps.
Yeah. They're,
Yeah, good
they
enough.
work good.
Yeah.
I mean, I don't hear myself echoing. I can hear you. Sure you don't hear.
That's why I took it off.
oxygen minimum is about the same depth as where this area is, so it's pretty common for there to be low oxygen. Oxygen minimum is at about 800 meters, so that is why the oxygen is so low at this site, because it is sitting at what is typically oxygen minimum area. as you can see in the aft camera, the vehicle is just about under the docking head. J-Rod's flying and kind of backs it up towards the ship. And then once it's right under the docking head, the winch operator will pull it out of the water. Cody's running the deck, so it's his call when that should happen. And so that's ROV out of the water. And that's the end of our dive today, guys. Thank you so much for joining us. This is Schmidt Ocean Institute, Research Vessel Falkor, and ROV Sebastian. We look forward to you joining us tomorrow, where we will be diving again on this site. We're just about midway between the coast of California and Catalina Island, off of Long Beach. So again, tomorrow we'll be hunting for DDT barrels or barrels of some type. Obviously we won't know until all the samples get back to the labs and all that kind of stuff. So